Melalui pembelajaran berbasis proyek, mahasiswa diajak untuk mengasah keterampilan berpikir kritis, kreativitas, dan kolaborasi dalam tim. Mahasiswa tidak hanya belajar teori, tetapi juga belajar bagaimana mengaplikasikan teori dalam situasi nyata. Fakultas Farmasi Universitas Setiabudi bekerja sama dengan berbagai perusahaan farmasi untuk memberikan pengalaman langsung kepada mahasiswa, sehingga mereka dapat memahami kebutuhan industri dan siap bersaing di dunia kerja.
Setiap langkah di Fakultas Farmasi Universitas Setiabudi Surakarta merupakan langkah menuju masa depan yang cerah.
Kami bangga menjadi bagian dari perjalanan pendidikan para mahasiswa, membentuk mereka menjadi farmasis, peneliti, dan profesional yang handal, edukatif, berbudi pekerti, aktif, dan terampil di bidang kefarmasian. Farmasi hebat!
Fakultas Farmasi Universitas Setiabudi telah bekerja sama, baik dalam negeri maupun luar negeri, seperti apotek, rumah sakit, PBF, industri obat, industri makanan, dan obat tradisional. Sedangkan untuk di luar negeri, Fakultas Farmasi sudah bekerja sama dengan beberapa universitas di luar negeri seperti Mahidol University, Rangsit University yang berada di Thailand, serta UITM di Malaysia. Fakultas Farmasi telah meluluskan berbagai lulusan yang menyebar seluruh Indonesia, yang membangun bangsa Indonesia, baik itu di Badan POM, Dinas Kesehatan, rumah sakit, industri obat, dan makanan. Mari bergabung Fakultas Farmasi USB bersama Fakultas Farmasi menghantarkan lulusan yang unggul, kompeten, yang mendunia.
Fakultas Farmasi USB hebat. H. Handal. E. Edukatif. B. Budi pekerti. A. Aktif. T. Terampil. Farmasi USB hebat, hebat, hebat.
Selamat datang di Universitas Setiabudi Surakarta.
Universitas Setiabudi Surakarta terletak di Jalan Ledjen Sutoyo Mojosongo, Kecamatan Jebres, Kota Surakarta, Jawa Tengah. Kampus Universitas Setiabudi Surakarta terletak di lokasi yang strategis dengan akses transportasi umum yang mudah dijangkau. Berdekatan dengan Terminal Tirtonadi, Stasiun Jebres, dan Stasiun Solo Balapan yang dapat ditempuh dalam waktu kurang dari 10 menit. Fakultas Farmasi Universitas Setiabudi merupakan perguruan tinggi pertama di Kota Surakarta yang didirikan pada tanggal 19 Desember 1995 yang merupakan salah satu universitas terbaik di Indonesia dan di Surakarta. Hingga saat ini, Fakultas Farmasi terus berkomitmen dalam memberikan pendidikan farmasi yang berkualitas dan inovatif. Fakultas Farmasi
Mohon
Universitas
perhatian.
Setiabudi
Dengan ini
menawarkan
kami
5
mengundang
program
hadirin
studi,
sekalian untuk
di
dapat
antaranya
segera menempati
D3
kursi yang
Farmasi,
telah disiapkan.
D3
Karena
Analisa
acara
Farmasi dan
akan segera
Makanan,
dimulai dalam
S1
beberapa
Farmasi,
menit lagi.
Profesi Apoteker, dan S2
Dan bagi hadirin
Farmasi.
yang baru datang dapat menempati dulu kursi paling depan.
Boleh untuk duduk di paling depan yang ada mejanya.
Disinilah tempat ilmu pengetahuan dan teknologi farmasi bertemu, menciptakan calon-calon tenaga kesehatan yang profesional di masa depan.
Saat ini, Fakultas Farmasi memiliki sekitar 1.800 mahasiswa aktif dengan didukung oleh dosen yang kompeten serta tenaga kependidikan yang berdedikasi. Hingga kini, fakultas ini telah menghasilkan lebih dari 14.000 alumni yang berkiprah di berbagai bidang farmasi dan kesehatan, baik di dalam maupun luar negeri. Didukung dengan tenaga pengajar yang berpengalaman dan kompeten di bidang kefarmasian, mahasiswa didorong untuk terus berinovasi, berkolaborasi, dan berkontribusi dalam penelitian yang bermanfaat bagi masyarakat.
Kuliah di Fakultas Farmasi Universitas Setiabudi memiliki kurikulum yang sudah dirancang dan diintegrasikan antara S1 Farmasi dan profesi apoteker, serta disesuaikan dengan Undang-Undang Kesehatan yang berlaku, sehingga dapat menciptakan lulusan yang berkompeten dalam bidang kefarmasian.
Bagi lulusan S1 Farmasi yang ingin melanjutkan studi ke jenjang yang lebih tinggi, Fakultas Farmasi Universitas Setiabudi memiliki program studi profesi apoteker dan program Magister Ilmu Farmasi. Lulusan S1 Farmasi USB mendapatkan prioritas untuk melanjutkan pendidikan di profesi apoteker USB.
Bagi lulusan D3 Farmasi yang sudah mendapatkan surat tanda registrasi tenaga teknis kefarmasian dapat digunakan untuk melaksanakan pekerjaan kefarmasian atau bisa melanjutkan jenjang studi ke S1 Farmasi melalui program RPL atau Rekognisi Pembelajaran Lampau.
Sementara, bagi yang tertarik kuliah di program studi D3 Analis Farmasi dan Makanan, lulusan D3 Analis Farmasi dan Makanan Universitas Setiabudi bisa mendapatkan pekerjaan bahkan sebelum diwisuda, atau juga bisa melanjutkan studi ke jenjang sarjana farmasi melalui program Rekognisi Pembelajaran Lampau atau RPL.
Fakultas Farmasi Universitas Setiabudi berkomitmen untuk membekali mahasiswa dengan pengetahuan dan keterampilan praktis di bidang farmasi melalui kurikulum yang terintegrasi dan berorientasi pada kebutuhan industri. Fakultas Farmasi Universitas Setiabudi memastikan setiap lulusan siap menghadapi tantangan global.
Namaku Laura Angelika, cita-citaku menjadi seorang apoteker. Karena apoteker memiliki peran penting untuk memastikan obat yang diberikan benar, dosisnya tepat, dan aman sesuai dengan kondisi pasien. Selain itu, apoteker juga memiliki peran dalam penelitian dan pengembangan obat-obatan baru. Saya berharap bisa menjadi bagian dari solusi untuk meningkatkan kualitas hidup pasien.
Fakultas Farmasi Universitas Setiabudi memiliki fasilitas laboratorium terpadu yang unggul dan modern untuk mendukung kegiatan perkuliahan, praktikum, dan penelitian di antaranya. Laboratorium Farmaceutical, Laboratorium Teknologi Sediaan Farmasi, Laboratorium Farmakologi, Laboratorium Kimia, Laboratorium Analisa Instrumentasi, Laboratorium Farmakognosi Fitokimia, serta Laboratorium Mikrobiologi. Fakultas Farmasi juga memiliki laboratorium unggulan seperti Laboratorium Teknologi Sediaan Farmasi, di mana mahasiswa dapat melakukan praktikum dan penelitian tentang pengembangan bentuk sediaan obat yang inovatif. Selain itu, Fakultas Farmasi juga memiliki laboratorium analisa instrumentasi yang dilengkapi dengan peralatan modern untuk mempelajari dan menganalisis komposisi kimia bahan baku farmasi. Serta laboratorium farmakologi yang mendukung penelitian mengenai efek farmakologis dan toksikologis dari obat-obatan. Dengan fasilitas yang lengkap dan modern, Fakultas Farmasi Universitas Setiabudi terus berupaya untuk mencetak lulusan yang kompeten dan siap menghadapi tantangan di dunia farmasi dan kesehatan, serta berkontribusi positif bagi masyarakat.
Mahasiswa di Fakultas Farmasi berasal dari berbagai daerah di Indonesia, dari Sabang sampai Merauke, dan dari Miangas sampai Pulau Rote. Universitas Setiabudi menjadi miniatur Indonesia yang melambangkan keanekaragaman dalam menimba ilmu di Fakultas Farmasi.
Halo, nama saya Wianda Rolani Elizabeth, asal dari Nusa Tenggara Timur. Saya sangat senang bisa berkuliah di Fakultas Farmasi Universitas Setiabudi. Di sini, saya bisa bertemu dengan banyak teman dari berbagai daerah, mulai dari Kalimantan, Sumatera, Sulawesi, Papua, NTT, dan banyak lagi dari daerah lainnya. Sehingga saya bisa mengenal dan mengetahui adat istiadat, budaya, serta bahasa daerah dari teman-teman kuliah saya.
Fakultas Farmasi Universitas Setiabudi Surakarta mendukung program Merdeka Belajar Kampus Merdeka. Mahasiswa dapat mengikuti program seperti magang, kampus mengajar, dan juga pertukaran mahasiswa merdeka.
Untuk mengimplementasikan Merdeka Belajar Kampus Merdeka, Fakultas Farmasi Universitas Setiabudi memberikan kebebasan dan fleksibilitas bagi mahasiswa untuk mengembangkan potensi diri. Melalui program ini, mahasiswa memiliki kesempatan untuk belajar di luar kampus dengan mengikuti pertukaran pelajar, magang di industri farmasi, atau melakukan penelitian untuk memperluas wawasan dan memperkaya pengalaman. Melalui pembelajaran berbasis proyek, mahasiswa diajak untuk mengasah keterampilan berpikir kritis, kreativitas, dan kolaborasi dalam tim. Mahasiswa tidak hanya belajar teori, tetapi juga belajar bagaimana mengaplikasikan teori dalam situasi nyata. Fakultas Farmasi Universitas Setiabudi bekerja sama dengan berbagai perusahaan farmasi untuk memberikan pengalaman langsung kepada mahasiswa, sehingga mereka dapat memahami kebutuhan industri dan siap bersaing di dunia kerja.
Setiap langkah di Fakultas Farmasi Universitas Setiabudi Surakarta merupakan langkah menuju masa depan yang cerah.
Kami bangga menjadi bagian dari perjalanan pendidikan para mahasiswa, membentuk mereka menjadi farmasis, peneliti, dan profesional yang handal, edukatif, berbudi pekerti, aktif, dan terampil di bidang kefarmasian. Farmasi hebat!
Fakultas Farmasi Universitas Setiabudi telah bekerja sama, baik dalam negeri maupun luar negeri, seperti apotek, rumah sakit, PBF, industri obat, industri makanan, dan obat tradisional. Sedangkan untuk di luar negeri, Fakultas Farmasi sudah bekerja sama dengan beberapa universitas di luar negeri seperti Mahidol University, Rangsit University yang berada di Thailand, serta UITM di Malaysia. Fakultas Farmasi telah meluluskan berbagai lulusan yang menyebar seluruh Indonesia, yang membangun bangsa Indonesia, baik itu di Badan POM, Dinas Kesehatan, Rumah Sakit, Industri Obat, dan Makanan. Mari bergabung Fakultas Farmasi USB bersama Fakultas Farmasi menghantarkan lulusan yang unggul, kompeten, yang mendunia.
Fakultas Farmasi USB hebat. H. Handal. E. Edukatif. B. Budi pekerti. A. Aktif. T. Terampil. Farmasi USB hebat, hebat, hebat.
Baik hadirin sekalian, karena acara sebentar lagi akan kita mulai. Untuk kursi yang masih kosong di depan dapat diisi terlebih dahulu. Bagi teman-teman yang masih di belakang, dapat mengisi terlebih dahulu untuk kursi yang paling depan.
Dan kami minta bantuan kepada panitia untuk mengarahkan peserta agar duduk di kursi terdepan terlebih dahulu. Kami meminta bantuan kepada panitia untuk mengarahkan peserta webinar untuk duduk di kursi terdepan terlebih dahulu.
Baik, untuk teman-teman yang masih ada di belakang, duduk di kursi paling belakang dapat berpindah terlebih dahulu untuk di kursi paling depan ataupun mengisi bagian tengah.
Assalamualaikum warahmatullahi wabarakatuh.
Shalom,
Om Swastiastu,
Namo Buddhaya,
salam kebajikan.
dan salam sejahtera bagi kita semua. Good morning ladies and gentlemen, we are delighted to welcome you to the first Setiabudi International Conference on Pharmaceutical and Health, or can be abbreviated as a first SBICPH.
Baik, selamat pagi hadirin sekalian. Kami ucapkan selamat datang di acara Seminar Internasional Pharmaceutical and Health pertama yang diadakan oleh Universitas Setiabudi Surakarta.
Yeah, however, before we start this international conference, first, we will greet all the attendees, ladies and gentlemen who have attended today.
Namun, sebelum kita memulai konferensi internasional ini, izinkan kami untuk menyapa terlebih dahulu seluruh hadirin yang telah hadir pada kesempatan hari ini.
To our honorable invitees, Dr. Laksana Saronsai from Raksit University,
Profesor Dr. Sabir Syekh Abdul, Medical Doctor, Master of Science, Dr. Philosophy from Taipei Medical University.
Nurainun binti Mohtar from University Teknologi Mara,
Dr. Apoteker Rina Herowati, Sarjana Sains, Magister Sains from Universitas Setiabudi Surakarta.
Dr. Siti Nur Fazilah binti Musain from University Teknologi Mara, and to Dr. Insinyur Joni Tarigan MBA as a Rector of Universitas Setiabudi Surakarta.
Dr. Randa Penny Pujiastuti, Magister Sains as Vice Rector 1 Universitas Setiabudi Surakarta.
Dr. Narimo Sarjana Teknik Magister Manajemen as a Vice Rector 2 of Universitas Setiabudi Surakarta.
Dr. Apoteker Titik Sunarni, Magister Sains as Vice Rector 3 Universitas Setiabudi Surakarta.
Bapak Agus Endrianto Suseno, Sarjana Ekonomi MBA, Vice Chairman of the Daily Management Yayasan Pendidikan Setiabudi.
Fahmi Mayasari, Sarjana Ekonomi, Magister Manajemen, Teacher of the Daily Management Yayasan Pendidikan Setiabudi.
Dr. Apoteker Iswandi, Magister Farmasi as a Dean of Faculty of Pharmacy Universitas Setiabudi Surakarta.
Dr. Apoteker Wiwin Herdwiani, Magister of Science as Vice Dean 1 of the Faculty of Pharmacy Universitas Setiabudi Surakarta.
Apoteker Francisca Leviana, Master of Science as of Dean 3, Fakultas Farmasi.
Swastika Ardana Riswari, Sarjana Ilmu Komunikasi, Kepala Biro Penerimaan Mahasiswa Baru Universitas Setiabudi Surakarta.
Profesor Dr. Apoteker R.A. Utari, Sarjana Utama, Magister Manajemen, Master of Science.
Dr. Apoteker Dian Marlina, Sarjana Farmasi, Magister Science, Master of Science, Doctor of Philosophy, as Chairman of the Committee.
And to our great committee and participants of today's international conference. Ladies and gentlemen, in Indonesia, we have an expression that say, tak kenal maka tak sayang. Which means if you don't know us, so you cannot love us.
Ya, jadi izinkan kami untuk memperkenalkan diri terlebih dahulu. I am Steven Barrera.
And I'm Yemima Aurelia.
As the Master of Ceremony, once again welcome to you to the first Setiabudi International Conference and Pharmaceutical and Health with Global Health Challenges in Pharmaceutical Research.
First, we would like to say the utmost gratitude toward God for letting us together today with good and stable state of health.
Alright, we will read on the rundown of today's event, which is divided into 5 parts.
First, opening ceremony,
The signing of the implementation of collaboration,
speakers presentation,
Q&A sessions,
and the last and with closing. Entering the first part, singing the national anthem Indonesia Raya and continuing with Universitas Setiabudi hymne. Ladies and gentlemen, please stand up.
Baik, memasuki acara yang pertama menyanyikan lagu Indonesia Raya dan dilanjutkan himne Universitas Setiabudi. Hadirin dimohon untuk berdiri. Hadirin
Attendees are welcome to sit back.
hadirin dimohon untuk duduk kembali.
Ladies and gentlemen, now we would listen to the welcome remarks. And the first welcome remark will be delivered by Apoteker Dian Marlina, Sarjana Farmasi, Magister Sains, Master of Science, Doctor of Philosophy, as a chairman of the committee. Dear Apoteker Dian Marlina, please welcome.
The Honorable Mr. Agus Endrianto Suseno, SA MBA, Mrs. Fahmi Mayasari, SA MBA, Daily Executive Board of the Setiabudi Educational Foundation. Dr. E.R. Joni Tarigan MBA, Rector of Universitas Setiabudi. Dr. Randa Penny Pujiastuti, MSI, Vice Rector 1 of Universitas Setiabudi. Dr. Narimo STMM, Vice Rector 2 of Universitas Setiabudi. Dr. Apoteker Titik Sunarni MSI, Vice Rector 3 of Universitas Setiabudi. Dr. Apoteker Iswandi SSI MPharm, Dean of Faculty of Pharmacy, Universitas Setiabudi, and all members of its structural management. Distinguished keynote and invited speakers, scholars, and all participants of the conference. Assalamualaikum warahmatullahi wabarakatuh. Shalom, Namo Buddhaya, Salam Kebajikan, Rahayu. Good morning, ladies and gentlemen. It is with great pleasure and honor that I welcome you to the first International Setiabudi Conference on Pharmaceutical and Health. Today, Saturday, January 25, 2025, marks a significant milestone for our institution as we embark on this inaugural event. The theme of our seminar, Global Health Challenges in Pharmaceutical Research, has been carefully selected to address the pressing health issues that transcend broader and affect communities worldwide. In an era where health crises can emerge rapidly and unexpectedly, it is imperative that we, as members of the global scientific and medical community, collaborate and innovate to find the solutions. This theme underscores our commitment to exploring the pivotal role that pharmaceutical research plays in tackling these global health challenges. We are honored to have an esteemed lineup of speakers today, including our keynote speakers. We have Dr. Laksana Charonchai from Rangsit University, Thailand. We have Professor Dr. Sabir Syed Abdul, medical doctor, Magister of Science, Doctor of Philosophy, from Taipei Medical University, Taiwan. We also have Dr. Nur Ainin Binti Mohtar from University Technology Malaysia. We also have Dr. Apoteker Rino Herawati, MSE, from Universitas Setiabudi, Indonesia. Here we also have our invited speaker, Dr. Siti Nur Fazila from University Teknologi Mara, Malaysia. Their expertise spans various fields of pharmaceutical and health sciences, and we are eager to gain insights from their extensive research and experiences. Ladies and gentlemen, the seminar is conducted in hybrid format, accommodating both offline participants here at our venue at Solia Zigna, Surakarta, and online attendees joining us from Zoom, webinar, and YouTube. This approach not only broadens our reach, but also exemplifies our adaptability in technology to foster knowledge exchange. I am pleased to announce that today's event has gained significant interest, with 408 participants attending offline and 572 participants joining us online from various regions. Furthermore, we also schedule about 102 oral presentation participants. The outcomes of this conference are substantial. We aim to disseminate the knowledge shared here through publication in several journals. We will publish in ISBN conference proceeding, the Cinta for Accredited Journal Pharmacy of Indonesia, the Journal of Pharmaceutical Research. Notably, selected article will also have the opportunity to be published in Scopus Index Journal Asset. Our distinguished guests, this event would not have been possible without the unwavering support of numerous individuals and organizations. I extend my deepest gratitude to the Setiabudi Surakarta Educational Foundation, our esteemed rector, vice rectors, deans, head of the departments, the seminar committee, our colleges, students, and the various vendors and sponsors who have contributed to bringing this conference to fruition. In closing, I encourage all participants here to actively engage in the discussions, ask questions, and network. Let us use this opportunity to learn from one another and build partnership that will extend beyond this conference. Might the knowledge exchange here today not only benefit for individually, but also contribute significantly to the advancement of science and technology globally. I am confident that together we will make a meaningful impact on the future of global health. Thank you for your attention and participation. Wassalamualaikum warahmatullahi wabarakatuh.
Baik, terima kasih kepada Apoteker Dian Marlina atas sambutannya. The second welcome remark will be delivered by Dr. Apoteker Iswandi, Magister Farmasi as the Dean of the Faculty of Pharmacy, Universitas Setiabudi, Surakarta. Dear Dr. Apoteker Iswandi, please welcome.
Okay, thank you. Good morning, everyone. The Honorable, the Executive Board Education Foundation Setiabudi, Mr. Agus Indrianto Suseno MA, and Mrs. Mahfi Maisari SMA. The Honorable Rector Universitas Setiabudi, Dr. Insinyur Joni Tarigan MBA, Vice Rector Academic, Dr. Penny Pujasuti, Vice Rector Research and Career Development, Dr. Apotheker Titik Sunari, the Honorable Vice Dean Academic Faculty, Dr. Apotheker Wiwin Herdiwani, Vice Dean Finance Faculty Pharmacy, Dr. Apotheker Ismi Rahmawati, the Honorable Vice Dean Student for Alumni Affairs Faculty Pharmacy, Apotheker Francisca Levena, and from the Honorable our speaker, Dr. Laksana Kontai from Rangsit University, Thailand. Welcome to Solo. This is the first time you come to Indonesia. Okay, the Honorable from the Prof. Dr. Sabir Syed Abdul Mat, MCC PhD. This is online from Taipei Medical University, Taiwan. Ms. Ainun Binti Mukhtar from University of Technology Amara. Welcome to Solo. This is second to Solo, right? Yes, thank you. And then Dr. Apotheker Rina Herawati from the Setiabudi University, and invite speaker Dr. Siti Nufardila from UITM. Thank you very much. And the Honorable former Dean, Professor Dr. Era Utari, which has become to my conference. Assalamualaikum warahmatullahi wabarakatuh. Om Swastiastu, Namo Buddhaya, Good Ways, Salam. First of all, I'll express the gratitude forward the committee who was blessing the world with us. The will all can be here in good condition and health. It's such an honor to give me welcome speech the first Setiabudi International Conference on Pharmaceutical and Health. The second time, the first conference is Global Health in Pharmaceutical Research. For us to know, global health is what research that has brought impact on the quality of life of people throughout the world. Research in the pharmaceutical field plays an important role in a safe, this goal, developing effective drugs and therapies of various diseases, both known and emerging. Innovation in pharmaceutical research include vaccine development, biotechnology-based medicine, and gene and stem cell therapies. Technology advances as artificial intelligence and big data analysis also play an important role in speeding up the drug research and development process. International cooperation is very necessary to face global health challenge. Global health organization, research institution, the pharmaceutical industry, and government around the world must work together to ensure the new discovery can be developed, applied to everything. On which joint effort can be achieved with the health of the humanity. We would like to express to great help the conference activity like this can be held every year. The event like this, we not only with knowledge, but also strengthen academic collaboration and expert professional network in the health and pharmaceutical field. We are very grateful for the support from the Setiabudi Education Foundation, the Rector and Vice Rector, and the extraordinary cooperation from UITM and Rangsit UTM and Taipei Medical University is the implementation cooperation integration in conference. This collaboration not only enrich from our insight and knowledge, but also strengthen academic relation between our institution. To the speaker, we would like to express to your side thanks for the time and dedication. The material present will be inspiring and provide new insight for all us. We would like to time to minister how hard the EI Education, the training agency, then all sponsor. Not to forget, we also want to the all participants, both online, offline, who activity practices. Hopefully, the knowledge gained today can be applied in daily activity, provide great benefit to material progress. To Dr. Insinyur Joni Tarigan, MBA, as a rector, would like to give speak and open this conference. Finally, once again, we would like to thank to all parties we had contribute the implementation of this conference. Hopefully, this collaboration will continue and have a positive impact on all us. Thank you for attention. Wassalamualaikum warahmatullahi wabarakatuh. Farmasi hebat.
Kami ucapkan terima kasih kepada Dr. Apotheker Iswandi yang telah berkenan untuk memberikan sambutannya.
And the last welcome remark will be delivered by Dr. Insinyur Joni Tarigan, Master of Business Administration as the Rector of Universitas Setiabudi. Also would officially open this event. Dear Dr. Insinyur Joni Tarigan, please welcome.
Assalamualaikum warahmatullahi wabarakatuh. Selamat pagi dan salam sejahtera buat kita semua. May peace and blessing be upon us all. Honorable guests, the Daily Executive Board of the Setiabudi Education Foundation, Mr. Agus Edriantono Suseno, all of file rectors, Universitas Setiabudi, the Dean of the Faculty of Pharmacy, Pak Iswandi, Profesor Dr. Apoteker Ibu Tari, distinguished speakers from local and international institutions, the chairperson of the International Seminar Committee and all members of the organizing team, distinguished scholars and all participants of the seminar. Good morning for all of you, my friend, and also my colleagues in these rooms. First and foremost, let us express our gratitude to the Almighty God for His countless blessings and graces that have enabled us to gather here today for the first Setiabudi International Conference on Pharmaceutical and Health, Global Health Challenges in Pharmaceutical Research. I extend my warmest welcome and highest appreciation to the Faculty of Pharmacy, Universitas Setiabudi, for organizing this first international seminar. These events serve as valuable platforms to share knowledge, exchange experiences, perspectives, and best practices among academics and experts from partner universities from different countries. Ladies and gentlemen, today we know that the world faces increasingly complex and urgent global health challenges. Chronic diseases such as diabetes, cancer, and cardiovascular diseases are on the rise, driven by modern lifestyle changes. Meanwhile, the threat of antimicrobial resistance has become major concern as many pathogens are developing resistance to existing drugs. In addition, the recent global pandemic has highlighted significant gaps in access to healthcare, particularly in developing countries. Multidisciplinary collaboration is key to addressing these challenges. The seminar provides an exceptional opportunity for us to share insights, exchange experiences, and strengthen synergies across fields of disciplines. Through such efforts, we can not only address global health challenges, but also create impactful solutions that benefit communities worldwide. Distinguished guests, I firmly believe that through intensive discussion and collaboration at this seminar, we can generate solutions that are not only innovative, but also inclusive and impactful. Let us make this international seminar as a starting point for integrating expertise and resources to achieve a better future for global health. Finally, I hope that the international seminar will contribute to the design of higher education governance policies based on the principle of good university governance and serve as a springboard for achieving excellence in accreditation. As a director of Universitas Setiabudi, I hereby officially open the first Setiabudi International Conference on Pharmaceutical and Health. I thank you for your kind attention. Wassalamualaikum warahmatullahi wabarakatuh.
Terima kasih kami ucapkan kepada Dr. Insinyur Joni Tarigan MBA yang telah berkenan untuk memberikan sambutannya sekaligus membuka acara kita pada pagi hari ini. The opening ceremony has ended. However, before we get into the main event, we will watching the signing of implementation of the collaboration between Universitas Setiabudi with Rangsit University and University Teknologi Mara.
Ya, rangkaian opening ceremony telah berakhir. Namun sebelum kita memasuki acara inti, kita akan bersama-sama menyaksikan penandatanganan implementasi kerjasama antara Universitas Setiabudi dengan Rangsit University dan University Teknologi Mara. Kepada panitia yang bertugas, kami persilahkan untuk dapat mempersiapkan prosesinya. Dan kami turut mengundang Dr. Anda Penny Pujiastuti, Magister Sains, dan Dr. Apoteker Iswandi, Magister Farmasi, untuk prosesi penandatanganan MOU.
And also we invite able to move forward to Durainun binti Mohtar from University Teknologi Mara for this signing of the implementation of the collaboration between Universitas Setiabudi and University Teknologi Mara. Yeah, thank you for Nurainun binti Muhtar from University Teknologi Mara. You can return to your seat back.
Dan kami himbau untuk Dr. Anda Penny Pujiastuti dan Dr. Apoteker Iswandi untuk tetap di tempat.
And now we invite to able to move forward to Dr. Laksana Charoncai from Rangsit University. With the signing of this implementation of collaboration between Universitas Setiabudi with Rangsit University and University Teknologi Mara, we hope that this collaboration can advance in the world of education and research.
dengan ditandatanginya implementasi kerjasama antara Universitas Setiabudi dengan Rangsit University dan University Teknologi Mara, kami berharap kerjasama ini dapat menjadi bukti nyata dari partisipasi kita untuk turut memajukan dunia pendidikan dan penelitian khususnya di kancah internasional.
And the next session is photo session. We invite all of the attendees can move forward for this photo session.
Baik, selanjutnya adalah foto bersama. Kepada panitia yang bertugas kami persilahkan.
Kami undang kepada rektor dan juga wakil rektor kami dapat maju ke depan untuk kita sama-sama mengadakan foto bersama.
Ladies and gentlemen, given that this event will not able to be held properly without the cooperation and support of main parties, therefore in the moment we will watch one of the sponsor video by PT Tisan Nugraha Globalindo.
Hadirin sekalian, mengingat acara ini tidak akan terselenggara dengan baik tanpa adanya dukungan dari pihak lain, karena itu di momen ini kita akan sama-sama menyaksikan salah satu video yang disponsori oleh PT Tisan Nugraha Globalindo. Selamat menyaksikan.
Selamat datang di PT Tisan Nugraha Globalindo. PT Tisan Nugraha Globalindo merupakan perusahaan jasa manufaktur kosmetik yang memproduksi kosmetik dengan kualitas yang tinggi. Kami memiliki sertifikat CPKP dan sistem jaminan halal. Kami memiliki tim research and development yang selalu berinovasi dan mempunyai pengalaman yang matang. Dibantu oleh SDM yang unggul dan sistem administrasi yang terstruktur, kami yakin dapat membantu Anda untuk mewujudkan impian Anda menjadi brand owner dengan berbagai produk kosmetik seperti skincare, body care, hair care, parfum, make up, dan lain-lain. Kami selalu melakukan quality control pada produk sebelum dikirimkan kepada Anda.
Bersama kita bisa with Tisan.
We are very grateful for the support from the collaborator, vendor, committees, lecturers, and students who have sponsored this event.
Kami sangat berterima kasih kepada kolaborator, vendor, panitia, dosen, dan mahasiswa yang telah mendukung acara ini hingga terselenggara dengan baik.
Baik, hadirin sekalian, tibalah kita di acara inti kita, yaitu pemaparan materi dari pembicara hebat kita pada hari ini.
Ladies and gentlemen, now it's time for us to enter the main part of this event, the presentation of material from our great speakers. First, Dr. Laksana Charonsai from Rangsit University with Analytical Quality by Design, AQBD, in Pharmaceutical Development.
Profesor Dr. Sabil Syekh Abdul, Medical Doctor, Master of Pharmacy, Doctor of Philosophy from Taipei Medical University with Artificial Intelligence and Digital Solution for Mitigating Global Health Challenges.
And Nur Ainun Binti Mohtar from University Teknologi Mara with Empowering Pharmaceutical Research, Synergetic, Role of Natural Product Development in Silico Tools and Quality by Design Framework.
And the last, Dr. Apoteker Rina Herawati, Sarjana Sains, Magister Sains from Universitas Setiabudi Surakarta with Exploring the Anti-Arthritis Activity of Indonesian Herb Medicine. Tapi sebelum itu kita akan mengundang moderator kita yang tidak kalah hebatnya. Dan berikut akan kami bacakan sekilas perkenalan singkat tentang beliau.
And this is a brief introduction about our moderator.
Baik, moderator pertama kita yaitu adalah Apoteker Dian Marlina, Sarjana Farmasi, Magister Farmasi, Master of Science, Doctor of Philosophy. Beliau lahir di Kabupaten Semarang pada tanggal 21 Agustus dengan riwayat pendidikan beliau S1 di Universitas Setiabudi, bidang ilmu farmasi dan lulus tahun 2013. S2 di Universitas Kristen Satya Wacana dan Kwangsai Gokin Universitas Japan, double degree dengan mengambil bidang studi biologi dan kimia dan lulus tahun 2016. Di S3-nya mengambil di Sekolah School of Human Development and Environment, Kobe Universitas Jepang dengan bidang studi kimia dan lulus tahun 2020.
Alright, now please welcome Apoteker Dian Marlina. Before we move to the main part, so this is the photo session with all attendees and all participants. Please stand up. Ya, untuk sesi berikutnya adalah foto bersama dengan partisipan. Baik, kami undang untuk hadirin, dimohon untuk dapat menempatkan diri untuk kita foto bersama terlebih dahulu. Yeah, we invite all of attendees, to honorable attendees, please stand up and we will have a photo session. Oh yeah, all participants can stand up too. Yeah, mari hadirin boleh berdiri. Okay, thank you everyone. You can stand up. You can sit back. Okay. All right, now we call back again. Please welcome Apoteker Dian Marlina.
Okay, we finally here in the main session. Ladies and gentlemen, distinguished guests and esteemed colleagues, welcome to the plenary session of the first Setiabudi International Conference on Pharmaceutical and Health. It is a great honor to have you all here today, both those attending in person offline and those joining us online. Today, plenary session is designed to provide a comprehensive exploration of current trends, challenges, and innovation in pharmaceutical and health sciences. We are privileged to host 3 distinguished speakers who will share their expertise and insight. In this session, we will have Dr. Laksana Charonchai from Rangsit University, Thailand. She will be presenting offline. And the second speaker, we will have Dr. Ainun from University Teknologi Malang, Malaysia, and also she will be presenting offline. And in the online session, Professor Dr. Sabir Said Abdul, Medical Doctor, Magister of Science, Doctor of Philosophy from Taipei Medical University, will joining by online. Okay, each speaker here brings a wealth of experience and a unique perspective to their respective, promising a session rich in information and inspiration. Okay, before we start, I would like to read the guideline for this session. So today, maybe for this session, I will use dual language. We can use English and Indonesia. So for all of the students here, please don't be worried to try to talk in English and also in Indonesia. We are here together to learn together and encourage all of you to the international environment. Okay, today's plenary session will be proceed as follows. Each speaker will deliver for 45 minutes presentation. They will talk about their specialized topic, sharing research finding, case studies, and practical applications. And following the presentation, we will open for about 45-minute Q&A sessions. This will be an opportunity for all of you to engage directly with our speakers, seek clarification, and discuss the implication of their presentations. We highly encourage active participants. You can ask any question or give some contribution here to discussion and learning. The organizing committee will arrange special prize. So ada hadiah spesial untuk teman-teman yang akan aktif bertanya di sini. Jadi kami harap teman-teman mahasiswa semuanya bisa aktif ikut serta dalam sesi ini untuk berlomba-lomba mencoba berinteraksi dengan pembicara kita, oke? Okay. And you can use both English and Indonesia, don't be worried, I'm here, ya. Okay. Okay, and to maintain a conducive environment for learning and discussion, please take a note of the following. Please kindly ensure that your mobile devices are set to silent mode to prevent any disruption during the session. And then those joining us virtually, please keep your microphone muted unless you are speaking during the Q&A session. And then if there are any technical difficulties, our support team is ready to assist. Please reach out via the chat function and we will address your concern promptly. Okay. Ladies and gentlemen, now let us proceed with the presentation. Beginning with our first esteemed speaker, we will have Dr. Laksana Charonchai from Rangsit University, Thailand. Okay, you can prepare your presentation and I will read briefly about your CV. So, assistant, associate or assistant? Assistant Professor Dr. Laksana Charonchai is a Drug and Herbal Product Research and Development Center from College of Pharmacy, Rangsit University, Thailand. She is the faculty member of College of Pharmacy, Rangsit University, Thailand. She is lecturer and researcher there since 2012 until currently, and lecturer in Faculty of Pharmacy and Science, Ubon Ratcha Chani University from August 2001 until December 2011. And then you are also has been the hospital pharmacist from May 1992 until 1995. And then also you visit Japan for research fellow, GSPS, October until November 2004. Also, you are joining ethical committee in Rangsit University, also laboratory safety committee. About her education, Dr. Laksana Charonchai finished the pharmacy in Bachelor in Mahidol University, Thailand in March 1992, and then continued to PhD in pharmaceutical science, medical chemistry, University of Maryland and Baltimore, US in May 2001. Her research area is analytical chemistry and medicinal chemistry, searching for active chemical and biological markers, analytical method development and formulation development of medicinal products. And about publication, she is active doing publication. This is so many publication. Up until now, the last publication is just 2024. Okay, without further ado, we will move forward with Dr. Laksana Charonchai. Today, she will share her insight on analytical quality by design in pharmaceutical development. Okay, ladies and gentlemen, please welcome Dr. Laksana Charonchai from Rangsit University, Thailand. Please, time is yours.
Good morning. Can you hear me very well? Good morning, everyone. And thank you for all the distinguished dean, guests, and also the seminar committee for inviting me to this conference. I would like to share with you about the review in analytical quality by design in pharmaceutical development. While waiting for the slide or the PowerPoint, the outline of my talk is about the drug development life cycle, process, analytical technology, and quality by design. And analytical quality by design. Then I will talk about the application and summary. Can I move to the slide? Yeah. Oh, okay. Yeah, let's go to the drug development life cycle. As everyone know about quality in pharmaceutical industry is the major concern for the consumer as well as the manufacturing and regulatory agencies. I would like to remind you about drug development life cycle. Scientists in the research lab, they discover or identify a new chemical entity or the new drug. After they get the new drug, or actually it's the new chemical entity, they will submit this to test as an in vitro or in vivo test. and the in vitro and in vivo test in animal or in the cell line, it will be to test for the safety and efficacy. And after that, they submit the investigational drug to the regulatory agency to approve for the human trial in the clinical research. So after preclinical, we go to the clinical research. Clinical research, typically there are 4 phases. The phase 1 is about the trial in small group of the volunteer, healthy volunteer, about less than 100 people to test for the dosage and the safety. Next, for the phase 2 trials in the medium amount of people to test for the efficacy and side effect. And after that, if the drug is the potential, they go to a bigger number, either healthy volunteer or the patients, to test for any further side effect. And after the clinical trial, next, they gather the knowledge or the information about the new chemicals and submit to the regulatory agency to review for potential efficacy and safety before granting it to the market. After they grant to the market and use in the in general people, this can go to the post-marketing surveillance in the phase 4. This one, after the drug used in the hospital or give it to the health provider, this medicine, since they discover is still high price, after use it for a while, maybe other company or competitor will introduce the generics or biosimilars after the patent expire. That will leading to reduce the price and market share. So in the process that we correct the knowledge, do the testing, the experiment, we need to control the quality of the medicine all over the drug life cycle. And the company may introduce new formulation or new indication to extend the product life cycle. Or the drug may be withdrawn from the market if we find any adverse effect. Or if this drug was replaced by the new version or the new treatment. So here, this is all of the drug life cycle. And next, I would like, this is the outline of my talk. And then next, I would like to give a little background about the history of quality by design before we go to the analytical quality by design. The concept of quality by design was created by Mr. Joseph Juran since 1970. After he developed and the US FDA implement this in the Good Manufacturing Practice or GMP, the regulatory organization or ICS, which stands for International Conference on Harmonization Technical Requirements for Registration of Pharmaceuticals for Human Use, released many documents about the quality of medicine or drugs. For example, ICS Q8 about pharmaceutical development, and this one was revised again in 2009, and then ICS Q9 is the quality risk management, ICS Q10 about pharmaceutical quality system, Q11 about development of manufacture of drug substance, and Q12 about pharmaceutical product life cycle management, And about analytical procedure development, two documents mainly mentioned in the Q14 and Q2 revised on the second time. These all documents you can access to the website ich.com if you interested in more detail. And next slide, I would like to go to about the process analytical technology, or we for short is the PAT, PAT. What's the definition of this one? ICH and FDA define it as a system for designing, analyzing, and controlling manufacturing through timely measurement during the processing of critical quality and performance attributes of raw and in-process materials, and process with the goal of ensuring final product quality. ICH also define this process analytical technology that the regulator shifts the control of product quality toward a science-based approach that attempts to reduce the risk to the patient by controlling manufacturing based on knowledge of the process. And this guidance will facilitate innovation in development, manufacturing, and quality assurance by focusing on process understanding. For the past, as FDA defined this framework, it can be divided into three categories: process understanding, principles and tools, and strategy for implementation. So you need to understand the critical source of variability, and then the variability that managed by the process. And product quality can be accurately and reliably predicted over the design space. The principle and tools that used in this is multivariate tools for design data, acquisition and analysis, process analyzer, process control tools, and then continuous improvement and knowledge management tools. To do this, they need to form a paired team approach and joining and training and their staff and scientific and technical support. In the traditional quality control and quality assurance, we need to analyze the material and in process and sample. So are we going to analyze it? How are we going to sampling it? Can be at line, online, or inline. In the past, it will be at line. At line means that you take the sample, measure the sample, that remove it for the analyze, for testing. And then we're going to move to online and inline, which means that you measure the sample that diverted from the manufacturing process and can be returned to the process. And by quality by design concept, we try to control in the real time, so we can test the sample inline. The sample was not removed, and the method that analyze it can be invasive or non-invasive. This concept will shift the industry from end product testing to more proactive approach where the quality is built into product. Next, I mentioned a little bit about the regulation agency. It starts from the FDA. They can be US FDA or the FDA in your country that maybe implement this regulation, and European Medical Agency, and all the documents that I mentioned earlier in the ICH. And also the United States Pharmacopoeia released the document in USP 12 and 20 about analytical procedure life cycle. this is referred to the procedural life cycle approach, emphasize the importance of sound scientific approach and quality risk management for the development control, establishment, and use of analytical procedures. Now, let's move to the concept of quality by design. Quality by design is a systematic approach to development that begins with predefined objectives and emphasize product and process understanding and process control based on sound science and quality risk management. And what is the quality? The quality is a product that free from contamination and reproducibility, delivering therapeutic effects, and that claim on the label claim to the customer. So the high quality drug product is those products that consistently and reliability deliver clinical performance and other characteristics as they mention on the label or label claim that products are not contaminated and safe for the customer, for the consumer. And in the traditional approach, we sampling and test the product, the raw material, by maybe relies on the final product testing. So the limitation of this approach is quality assessment rely on the end product testing, and when we sampling is how we sure that the sample that we sampling are the representative of the entire batch. There may be constraint the ability of the test sample to adequate represent the variability of the entire batch, and the time is limit for the test that it's gonna be pass or fail test result, and the test procedure and acceptance limits are used to ensure pharmaceutical quality not well suited to the mass production environment. That's why we gonna implement the concept of quality by design. Before we go to that, I would like to show this slide to give you the example. For example, if you would like to do the analytical method development in the past, probably we use isocratic system or gradient system of the mobile phase. We do this as a stepwise approach, change one factor at a time, and then we see that we observe the peak or not, and there is adequate resolution or adequate selectivity or not. If not, we come back to change one factor at a time. So if we use the quality by design methodology, this will be several steps. The methodology is composed of the first step. You have to know the knowledge about the product and the process. For example, the drug substance, active pharmaceutical ingredient, and the drug dosage form, and including the excipient and manufacturing process. When you understand this, you will define or establish the quality target product profile. Is the design product characteristic, the efficacy, safety, manufacturing process. After that, you identify the critical quality attributes, and then identify critical material attributes or critical process parameters, and prioritize what the critical quality attributes. These all the process go to, you need to use the tool that called quality risk management and design of the experiment. Once you get the knowledge space, and then you will get the design space, and you optimize those parameters. and you will get a control state and use this method for the control strategy. Then we can consistently monitor the quality of the product by life cycle management and continuous improvement. I would like to emphasize a little bit what is the design space. The design space is the multidimensional and interaction of the input variables. For example, material attributes and process parameters that have been demonstrated to provide assurance of the quality. Let's go to the example of the critical quality attributes. The critical quality attributes is the properties or characteristics that should be within the appropriate limits or the distribution to ensure the desired product quality. For example, for drug substance, it can be the appearance, particle size, morphology, water content, residual solvents, organic impurity, inorganic impurities, heavy metals, assay or related substance. And for solid dosage form, for example, tablet or capsule, it can be the shape, the color, the diameter, the physical form, the flowability of powder before you make the tablet and capsule, the polymorph of the active ingredient, and also the thickness, hardness of the tablet, moisture content, disintegration time, dissolution, degradation products, and also microbial limits. For the lipid dosage form, in this case, I would like to give the example about the solution and the suspension. So the quality attributes that you need to consider, it could be the appearance, the color of the solution, taste, odor, is a clear solution or is the separate or turbid, the density, the dispersion medium, volume, light diffraction, aggregation time, viscosity, particle shape, polymorph, content uniformity, order of mixing, addition of the ingredient, pH of the dosage form, zeta potential, and also quality and quantity of drug substance and excipient. And next, we go to now implement the quality by design to the analytical method development. The definition of analytical quality by design that we're going to talk about refer to a systematic approach that apply in analytical method development and optimization to ensure the reliability, robustness, and quality of analytical method used in pharmaceutical analysis and also other fields. This, you can see the workflow is similar to the quality by design. Starting from, you need to know the knowledge. It means understanding your analyte, either a drug substance or drug product, the active pharmaceutical ingredient or excipient that you want to analyze by defining analytical target profile or ATP, and then identify method attributes or critical analytical attributes, identify critical method parameters, and perform risk assessment, and then do the design of experiment, establish the method operable design region, and use in the control strategy to monitor of the product quality and continuous improvement cycle. So the analytical method requires the development, validation, and controls just like any other product and process development activities. So let's move to the analytical target profile. What is the analytical target profile? It is a predefined objectives that stipulates the performance requirements for the analytical procedure. It outlines the method, intended performance characteristics, and quality attributes. It states the required quality of the results in terms of acceptable criteria in the measure and the maximum measurement uncertainty. So it should include definition of the analyte, the description of the analytical metrics, and the range or acceptable criteria, precision, accuracy for the reported value. This slide, I would like to mention or show the example of method of analysis and what could be the critical quality attributes of this. As all of you may know that we usually use the chromatography techniques, many techniques that used to analyze the sample. It could be gas chromatography, high performance thin layer chromatography, or HPLC method. Let's see the first one, gas chromatography. It could be the parameter that we set when we perform the experiment. It can be the oven temperature for the column, other setting, injection temperature, gas flow rate, sample diluent, concentration, or carrier gas. And for HPLC, high performance, it can be stationary phase. You can use either stationary phase, silica gel, or high performance stationary phase, which is small particle than regular stationary phase, and mobile phase, sample concentration and volume, equilibrium time that we need to consider, developing time, and the reagent that we spray after we develop the plate for the detection. For lipid chromatography, we need to consider the stationary phase as well, the type of column temperature, and dimension, the mobile phase, elution method, and can be isochromatic gradient, what the mobile phase ratio, organic modifiers and additives, and also flow rate, and what type of the detection that you're going to use. So next, I would like to mention about the tool that you're going to use in the analytical quality by design. First, let's see the quality risk management. In the ICH Q9, they mention about the quality risk management. What is the risk? Risk is the combination of the probability of occurrence of harm and the severity of that harm. And quality risk management is a systematic process for the assessment, control, communication, and review of risks to the quality of the reportable value across the life cycle of the analytical procedure. This diagram shows the relation about risk assessment and risk control and the risk review, and after that we're gonna decide that we will accept or decrease, how we're gonna decrease the risk. And this quality risk management supports scientific and practical approach for decision making. And the risk management methodology that we use, it could be a flow chart, a heat map, like a process mapping, or a cause effect diagram, for example, Ichikawa fishbone diagram, or the FMEA, which stands for failure mode effects analysis. This one we will see the failure mode that established and see how gonna do the risk reduction to eliminate it. But for the FMECA, this one extends to the severity of the risk and of the consequence of it. Now let's move to the risk assessment. Risk assessment is the systematic process of organizing information to support risk decision to be made within risk management process, consisting of risk identification and risk analysis and risk evaluation. Risk identification, you need to identify the potential source of the harm, and after that you estimate the risk that associated with the identified hazards, and then compare the estimate risks, use qualitative or quantitative to determine the significance. After you do this assessment, you can decide that how you're gonna accept the risk or reduce the risk, and then share this information to your party, to your colleagues, to the industry and regulation. This slide shows the risk assessment metrics that you can define or identify, estimate it like a value of the probability to occur that harm and the consequence or the severity of that harm. And then you can use the number to estimate the severity or the significance of the risk, and see that how we're gonna reduce it. And this slide, I would like to give some example. When you want to define the critical quality attribute or critical process parameter, you need to evaluate the variable effects that the most impact on the procedural performance. In this slide, show the example of the, if you interested to analyze the active pharmaceutical ingredient separate from other impurities, or what impurity that cannot be separate, and with what is the factor. The factor could be the flow rate, the gradient slope, percent organic solvent, column temperature, or pH value of the mobile phase. And the response that we want to monitor is the resolution or selectivity or efficiency, that we can separate those two peaks or not. We can set the criteria and do the mathematics or statistic analysis. The criteria could be the resolution for those two peaks. For example, criteria for active pharmaceutical ingredient and impurity resolution should be higher than 3. And for other pairs of impurity, it can be higher than 1.5. This is just for the example. Next, we go to see the example of the fishbone diagram. Fishbone diagram is to see the cause and effects diagram. In this slide, show the example of the chromatographic analysis. What could be the factor that will have effect on the response? It could be men or people who analyze, the machine, the environmental of the laboratory, the method that you're going to use, the measurement, what the parameter that you need to measure in the analytical method that mentioned in the ICH guideline Q2, and the material, what the grade of the standard that you're going to use, and the sample metrics, and the techniques of the analyst. Another example is the more specific to liquid chromatography, that HPLC method. In similar, you can see the factor that affect. It could be material, mobile phase, instrument parameter, sample, column, and detection. So anything that can be the critical process parameter that we need to monitor For example, the sample, you should consider the pKa value, solubility, polarity, and the material that going to use is the standard, is working standard or reference standard, for example. An instrument that you're going to choose, what could be the parameter of that instrument. And now we're going to move to the design of the experiment. The design of the experiment, we use like a software or computer program to design this. The definition of this is the organized structure method for determining the relationship between factors that affecting a process and output of that process. This can be helped to identify the optimum condition instead of we change stepwise or one factor at a time. So this will be determine the process and the relationship between input parameters and output response, and use the computational design and statistics analysis. For example, full factorial design. They explore every possible combination of independent variables, or you just screening is systematic experiment by eliminating non-significant variables and then reduce the total number of the experiment. It can be fractional factorial design or Plackett-Berman design or Takeuchi orthogonal array design. Or some research, they use a response surface methodology. It's the optimization phase, focusing on key variables and their interactions, identifying the previous screening design. You can use like the full factorial, central composite, or Box-Bengen, or Dewhurst design. And this slide kind of summarize the idea or the concept of design of the experiment, that we have the input variables and we have the output, observed response variable. So this is similar to critical procedure or critical process parameters. And this is critical quality attributes that we need to define. In the analytical procedure, we need to know the procedural variables that we mentioned in risk assessment, material property, that we understand the product and process. For example, we can put this in the mathematics model, that the input variable, it could be some stationary phase and solvent composition, something like that. And this is the response that we're going to monitor, like peak symmetry, peak area, resolution. And this slide, I would like to mention about the method operable design legend. This one is the, after you got the knowledge space, design space, and control space, you will get acceptable performance region. That is the region that you can make a change in this when you monitor the quality of the drug product. Analytical change management, if any change occur in the analytical condition within this control space or in the MODR or method operable design region, this change does not require additional experiment before implementation. But if the change of other parameter outside, it may be require a risk assessment. Go to review that before we get the analytical method development. Now let's see that this is some example of the commercial computer software that used in the design of experiment. It can be the design expert, fusion quality by design, and dry lab and other software that come together with the instrument that you use in the analytical. There are maybe some other software. If you interested, you can go to see in the website. And this slide, I would like to compare a little bit about the quality by testing and quality by design approach. As we mentioned earlier in this talk, the quality by testing is from stepwise time to time, but quality by design, you build into a product systematically process design. And the process development in quality by testing is unique variate, but in quality by design is the multi-variate experiment. And this can fit in the manufacturer, cannot change easily. But if you use the quality by design approach, it can be adjustable within the design space. And you can focus only reproducibility, that you manufacture the product and you get consistent of the quality result. But in quality by design, you need to focus on the robustness and understanding and controlling of the variability. And this quality by testing can be process delay, but quality by design, it needs a decision team-based. And you can, in the traditional, you can use the in-process quality control. But for quality by design, it's a process control and feedback. And then you come back to review a risk assessment and then do the continual method monitoring or continual improvement. And this one, that for the quality by testing, it could be a narrow operating range, but quality by design, when they give you a design space, you got a control space and method operable design region that will rely on real-time quality testing. And the product performance is designed by the product specification. But in traditional and quality control, probably it depends on the batch control. So any change in the method operable design region, you can do maybe without post-approval after make the change. Now let's move on to the implementation and application of the quality by design. There are many articles that do the research and study this. They use this concept in the chromatographic techniques, spectroscopic measurement, and even in the hyphen techniques like LCMS, GCMS, and in bioanalytical method development and dissolution study, and in formulation development and biopharmaceuticals. In similar, analytical quality by design used to determination of the main components either in bulk or in pharmaceutical dosage form, and determine the multi-components in combined products, and determine the impurities, related substance, and degraded product, and also sample in various metrics, for example, in biological fluid, or even in the medicinal plant extracts. Let's go to one example. The first example, that they use quality by design approach in the HPLC method. This research, they describe about the risk-based HPLC method development and the validation of ceftriaxone sodium in pharmaceutical dosage form. Ceftriaxone sodium is one of the antibiotics. And in this research, they perform the experiment design using design expert software. based on the central composite design. So instead of change one factor at a time, they do the systematic experiment in 11 experiments by monitor the two factor, mobile phase ratio and the pH of the buffer in 3 levels. You can see that once you perform the experiment, you will get the equation. This one example of the equation, AB is the factor that you monitor. In this case, X is the acetonitrile to water mobile phase ratio. It can be 25, 75, 30, 70, 35, 65, something like that. And pH of the buffer in 3 levels, 5.5 to 7.5. After you use the, put your result in the equation and use the computer and statistics, you get equation and you get like a 3D plot or surface plot. And you can see that which factor that affect the retention time and theoretical period and peak asymmetry. When you decrease this value, increase this value, how it affects the retention time and theoretical period or peak asymmetry in the surface plot. And after that, you will get the optimized condition in this surface plot or contour plot in 3D diagram, and you perform the experiment and get a robustness of the method and get the optimal condition to analyze the main active pharmaceutical ingredient. Let's go to the second example, that they implement analytical by design or quality by design to determine the nitrosamine. You know the nitrosamine. This one I take the example from 2 articles that performed by the same research group. In the past, they found that nitrosamine contaminate in the drug product. In this case, it's sartan drugs or vasatans. And this drug, they use active pharmaceutical ingredients from the suppliers. And during the synthesis of these active pharmaceutical ingredients, there are some side reactions that can cause nitrosamine, and the nitrosamine will found to contaminate, resulting in we need to withdraw this product from the market. And let's see, what is the nitrosamine come from? In this reaction is the tetrazone ring formation step. This is one step in the formation of the vasatan. This is the tetrazone chemical structure. In this reaction, they need the catalyst, sodium azide, and zinc chloride, and solvent dimethylformamide with the sodium nitrite in acid condition. The azide reacts with a nitrite under zinc chloride catalysis in the dimethylformamide solvent. The sodium nitrite, the excess of sodium nitrite used to quench the excess of azide. So nitrosamine comes from the dimethylformamide that change to dimethylamine, and this react with the sodium nitrite and resulting in N-nitroso dimethylamide. That's why active pharmaceutical ingredient contaminate with the nitrosamine. So this group, they try to analyze active pharmaceutical ingredient simultaneously with the nitrosamine impurities. So they study the parameter using quality by design and use the software fusion quality by design and the analytical target profile that they're interested in to detect impurities individually and simultaneously with the sensitivity and separate all the other impurities, and the method should be complied with the regulators. They define the. Okay,
I'm sorry, your time is limited.
Yeah, okay,
Yeah.
the time is
Just
limited.
finish it.
I will try
Yeah.
to finish that.
Yeah.
Okay.
I'm so sorry.
And this one, they get define the critical attribute and critical quality attribute, and then the critical method parameters. After that, they do the experiment and get the result, and they can eliminate one factor from the eluent test. They got that the methanol can be the best eluent, and they vary trifluoroacetic acid and found that 0.1% trifluoroacetic acid is better in the analytical condition. They get the design space and method operable design reagent. And this group, they extend the analytical method for the continuous life cycle. First, they do the analytical method development to determine nitrosamine impurity in Wassertan drugs. So they extend to determine in outer drug that has similar synthesis method that has the process to form the tretazone ring that could be cause the nitrosamine impurity also. They perform the experiment, revise the analytical target profile, and then get a design space and method operable reagent, and finally get the optimal condition that can separate the impurities. And when we go through this workflow, we can keep continuous improvement the life cycle. As I mentioned earlier about the workflow of the analytical quality by design, they can use this as a set of control in the control strategy to ensure that the process performance and product quality is still consistently. And this come to the nearly wrap up about the analytical procedure life cycle that they mention all the steps in analytical quality by design and continuous improvement in the ICH Q14 and also in the USP that mainly three states, procedure design, procedure performance qualification, and ongoing procedure performance verification to continue improvement and monitor the quality of the drug through the manufacturing process. And again, this is one of the stage two that we need to ensure that all activities that perform confirm that the procedure is fit for the intended purpose that we set in the analytical target profile. And the advantage of quality by design concept that we implement is to increase the product and process understanding. And this is more flexible for life cycle management of analytical procedure chain and can reduce the variability and facilitate the continual improvement. However, there's still the challenge of the quality by design and analytical quality by design that it could be the internal misalignment in the industry that you should decide who gonna responsible for this, and lack of the technology or maybe lack of tangible guidance which can manage by proper framework and appoint the quality by design expert, ensure that you collect all knowledge, understanding the product and process, and develop, use the maybe commercial software, and maybe develop collaboration. And in summary, process analytical technology integrate in the modern pharmaceutical development to define systematic approach to process understanding, control, quality assurance, and leading to safer and more effective pharmaceutical product. And quality by design transform pharmaceutical development into science-driven, risk-based approach, enhance the quality, safety, and efficacy of drug product. And analytical quality by design play an important role in pharmaceutical development by providing structured approach, developing analytical methods, understanding material attributes and process parameters to drug product, critical quality attributes, ensuring product quality and facilitating regulatory compliance. And finally, quality should be built in the product. That's all of my talk. Thank you for your attention.
Okay, thank you very much, Dr. Laksana Charonchai, for your insightful presentation. It's really nice topic. We learn here about QDB. I want to check our students here. How are you guys? Fine. Okay. Anyone want a question? Nah, wait. We will still have two presenters here, so we will have Q&A session after all of the speakers talking today, okay? You can have a seat. Yeah, please. Once again, thank you very much, Dr. Laksana. Your discussion on QDB has enhanced our understanding to develop robust analysis method that ensure consistent performance throughout the product life cycle. Okay, now we will move to our second speaker, Dr. Nur Ainun from University Technology Mara, Malaysia. I will read her curriculum vitae briefly. So, Ibu Nur Ainun is a senior lecturer in University Technology Mara. He's doing now here doing her PhD in Malaysia Japan International Institute of Technology, MGIIT, UTMKL from up until now. Her previous position is lecturer in University Technology Mara, young lecturer from 2011 until 2013. Okay, for academic background, she got Master of Science in Bioprocess Engineering in University Putra Malaysia Selangor from 2009 until 2013. And then currently doing PhD in pharmaceutical technology. Her research focus and interested is in the computational chemistry, pharmaceutical and medicinal chemistry, bioprocessing, nanomaterial, nanotechnology, quality by design, QDB in biopharmaceuticals, process optimization study, synthetic biology department, and natural product development. About her publication is also, she's really active in the publication. You can check her publication in Google Scholar. You can check her Scopus. She is currently doing the research in natural product, right? Okay. And her last publication is in 2024. Okay. Today, Ibu Ainun will discuss about empowering pharmaceutical research, synergistic role of natural product development in silico tools, and quality by design framework. Okay, ladies and gentlemen, without further ado, please welcome Dr. Ainun binti Mohtar from University Mara, Malaysia.
Okay, Assalamualaikum warahmatullahi wabarakatuh and very good morning. Thank you, Dr. Dian, as our moderator today. Okay, how are you, everyone? Okay, I am quite worried because previously you have been introduced to something very technical in the very morning. Okay, and luckily I'm going to repeat that again. Unfortunately, I am sorry because it was in my, actually we have similar topics, okay? But I will try my best to reduce the tension, okay? I will try my best to deliver it as light as possible, okay? Please bear with me for another 45 minutes. All right, maybe we can go to my slides. Okay, just open the link that was given in the Canva. So I will control from my iPad here. Okay, but before that, before they are setting up for the slide, may I know, I just wanted to roughly to understand my audience today. So how many of you are in the first year of your study? Second year? Please raise your hand upright so I can see you. Okay, how about the third year students? Okay. All right, do we have postgraduate students here? How many of you postgraduates? Okay, so the rest of you, where are you from? You're not responding. Okay, maybe can I mix my speech today in both languages? Okay, in bahasa Malaysia lah, because I'm from Malaysia, right? Bahasa Melayu lah. Okay, and English because I can see the tension here. Is my slide available? Sudah bisa belum? Huh? Bagi link Canva, just open the link Canva. Okey lah, kat sini berapa ramai ya? Saya nak tanya. Ha? Okay, saya nak tanyalah berapa ramai yang dia rasa dalam diri dia, dalam jiwa dia, ada satu semangat, seru kita panggil seru, seronok. Kan, seru means happy, right? Semangat apa? Semangatlah, samalah kan, semangat kan? The burning sense, the burning feeling, that you want to create something good for the community. Okay. Please raise your hand. Tak ada juga. Okay. Tak ada juga, tak apa. Dah belum? Dah boleh? Tak boleh? Lama pula lama saya buat slide ni tau. I spend my endless night doing the slide. See? Beautiful kan? Okay. Alright, so hari ini saya akan share a little bit about my, nak kata expertise tu tak jugalah, but what I know, what the little things that I know that I can share with all of you today. And hopefully, based on my speech today, talk lah, bukan speech lah. Okay, based on my sharing session today, I hope you can gain something. Tak banyak, sikit pun jadi. The key message is important. Alright, so hari ini tajuk kita bunyilah gempak. Gempak dalam bahasa Indonesia, what we call it? Bombastic is gempak lah in Malaysia, means like something, wah, the title, okay? I have to ask ChatGPT to give me the title. Okay, so thank you to ChatGPT as well for today. Alright. Okay, before that. Okay, let me ask you, can someone, boleh tak dekat sini siapa-siapa bagi satu contoh global. Kita hari ini kan tema kita kan Global Health Challenges. Okay, boleh bagi satu challenge tak? Dah kita ni ya je nak cakap pasal challenges. Challenge ni tahu tak challenges tu apa? yang dalam pharmaceutical sekarang ni? Can you name me one? Satu je lah. Maybe someone can raise, just raise. Cabaran sekarang ni kan tema kita hari ni adalah kita nak menangani cabaran. Tapi kita tahu tak cabaran yang kita nak tangani itu siapa musuh kita sekarang ni? Kan? Okeylah. Katalah salah satunya apa? Microbial resistance. Betul? Suddenly. Tadi tanya semua senyap aja. Okay? Alright. So actually challenge ni banyak sangat challenge. Okay, kalau kita nak cerita pasal challenge sampai ke kiamat pun mungkin tak habislah. Okay? So untuk tidak, dia tak bergerak. Kenapa dia tak bergerak? Sebab saya tak present. Betul ke? Saya dah present. Sepatutnya saya boleh control dari sini. Ke you control? You tekan tepi. Oh, penat je. Okay, apa next? Alright, so sebelum the challenge, cerita dulu musuh kita. Okay, the challenge is the infectious disease, of course, as well as the bacterial resistance. Yang ini memang never ending story. The fight has never been resolved lagi. Tak habis lagi berperang. I'm sorry, Dr. Laksana, I have to talk in Malay language. Okay, please excuse us. Okay, and the second one, we also have mental health. Okay. This is a current challenge that we need to address and acknowledge. At the moment, this is one of the top global health challenge. Next, we also have non-communicable disease. For example, apa? Diabetes, cancer, the top disease lah. And then malnutrition and as well as food security. Okay, next please. I thought I can. From here, I cannot. Okay. All right. So, what are our strategy in addressing this? Okay, one of the way is we need to strengthen. Okay, we need to strengthen the research ecosystem. And how? How do we do it? Okay, maybe we can implement. This is the time to implement the innovative drug development. We cannot, like traditionally, we do the try and error. Okay, try and then fail and then repeat again the process. This is not the time. We need to be more innovative in drug development. So we need to strengthen by cross-disciplinary collaboration. Okay, another one is maybe, we can advance the natural product research. Why natural? Why not synthetic? Synthetic is much easier, right? We can like create something in the lab and proceed. But why? Why do we need to start with natural? We're going to look at this later. Okay, next. Okay, and then since we are already in a Industrial 4.0, as well as Pharmacy 4.0, and what does it mean? It means that we need to leverage technology, for the drug discovery. Okay, then since we have all these resources, it doesn't mean that with all the technology that we have, it doesn't mean that we need to be stagnant. We need to optimize. Okay, whatever the tools we have, apa sahaja resources yang kita ada, kita perlu optimizekan. Walaupun kita ada teknologi, tak semestinya teknologi itu adalah yang optimum. Jadi apa yang kita perlu lakukan? Kita perlu optimizekan whatever resources yang kita ada. Okay, dan itulah di mana yang Dr. Laksana Dr. Laksana dah share dengan kita begitu detail sekali tentang QBD, quality by design. Because quality by design, these days we cannot waste our resources anymore. It's not the era that we have abundant of resources. Kita kena optimizekan whatever yang kita ada sekarang ni, even the smallest amount, kita juga boleh proceed with the bigger impact, provided that we optimize the resources. And that is why QBD can help us, the tools that we can use. Okay? And then we need also to embrace sustainability, okay, in pharmaceuticals. What does it mean? Sustainability means walaupun kita ni begitu gairah untuk mencari, explore research ni, mencari solution, tapi kita jangan lupakan tentang implikasinya juga terhadap environment. So kita kena make sure whatever kita kena bertanggungjawab jugalah, okay, and maintaining the sustainability in pharmaceuticals. Okay, there is no point kalau kita begitu apa orang kata advance in producing drugs, tapi the drugs itself cannot be sustained in the future, or it can give a negative impact to the environment. So we need to balance. Okay, and another important parts of addressing the challenges in pharmaceutical is we need to have a global collaboration. Kita tak boleh bergerak orang yang sama dalam satu bidang sahaja. We need to have few heads, few ideas. Okay, barulah sesuatu tu boleh jadi holistik. Sebab macam contoh ya, kita ni kadang kita rasa orang farmakologi, okay, they have their own perspective. Okay, dia hanya nampak, dia punya tumpuan dia tertumpu kat situ. Tapi orang di dalam bidang industri ataupun orang di dalam bidang manufacturing, dia tak nampak. Dia tak nampak apa yang orang pharmacological ni nampak. Jadi that is why we need to have all input. Okay, we need to have all inputs from various field. Okay, and do collaboration, especially within the academic, industry dan juga society lah. Okay, next. Okay, so the strategy yang hari ini saya nak bincangkan fokus kepada 3 area, iaitu natural product design and exploration, and how this natural product can be leveraged by implementing the QBD framework and harnessing the technology. Okay, sebelum ni biasa dengar in vitro kan? In vitro, in vivo. Okay, how many of you biasa dengar in silico? Siapa first time dengar in silico hari ni? Biasa dah dengar. Ada yang biasa, ada yang first time. Okay, in silico just a simple, dia nak bagi nampak senada dengan in vitro dengan in vivo lah. Okay, so in silico means it is a computational in silicon. Okay, sebab sekarang kita dah zaman AI kan. So zaman AI kita dah start ada various machine learning, okay, AI, artificial intelligence to help us in the pharmaceutical research. Okay, next. Okay, so kenapa natural product? Do you believe me if I say 70% of the approved drugs these days yang kita pakai sekarang ni, yang kita rasa sintetik tu sebenarnya berasal daripada natural. Okay, yelah, it's a logic thing. Sebab, kenapa? Sebab manusia, dia akan tengok alam sekeliling dia. Dia mencari solusi di sekeliling dia. Solusi yang paling dekat dengan dia adalah natural. Betul? Okay? Natural. Jadi sebenarnya even the antibiotics, especially antibiotics, 100% of antibiotics is actually derived from the knowledge of natural product yang daripada microorganism lah. Okay? Biasanya daripada microorganism. So bila kita sebut pasal natural product, dia tak semestinya pokok saja. Okay? Kita ada various type of natural resources. Okay, for example, kita ada plant resources yang kita biasa tengok lah. And then animal resources juga dikira sebagai natural product. Can you name me? Animal punya drugs yang derive daripada animal. Ataupun contoh animal yang kita gunakan. Never mind. You find the answer. I give you time. Okay. Next, microbial resources. Okay, microbial resources macam tadi lah, especially untuk develop antibiotik. And juga kita ada mineral resources. Next. Okay, so bila kita kata kita nak start dengan natural product, macam mana kita nak mulakan? Okay, by standard lah, biasa kita start identifying the plant yang kita berminat, ataupun kita jalan-jalan, cantik pokok ni, agak-agak apa dia boleh buat? Kan? Kadang-kadang kan kita terfikir macam tu kan. So kita start dengan tengok dan kaji pokok tu. Okay, so we do the extraction lah. Itu biasa kan proses dia. Kita do extraction. Lepas tu kita check dia punya kromatografi. Apa ada dalam ni compound A, B, C, D, E, F, G. Lepas pada tu kita try juga. Antara compound ni A, B, C ni yang mana paling okey. Okay, yang mana yang function. Kadang-kadang dia ada aja tapi tak ada function pun. Sama juga. Macam sebab tu sebaik-baik manusia adalah manusia yang bermanfaat kepada manusia yang lain. Macam tu. Sama juga pokok ni. Tak ada reason dia ada diwujudkan dalam alam fana ini kalau dia tidak ada function. Okay, so mesti ada function, tapi function yang okey ke tak okey ke, applicable untuk human ke tak ke, itu yang kita kena kaji. It's left to us to find out. Okay. Dan daripada situ, lepas kita dah identify compound dia, kita try pula tengok assessment. Okay, dekat dalam praklinikal. Tadi ada berapa clinical test? Ada berapa fasa? 4. Kan? Okay, ni pay attention tak ni tadi? Okay. Alright, next. Lepas tu, barulah kita boleh submit untuk approval dan digunakan untuk manusia. Next. Okay, ni antara contoh. Okay, saya rasa kamu pun macam dah tengok gambar pun macam never mind. I think the community can share the link for this canvas. So later on you can go back and study. Study apa benda lah yang dia ni cakap ni kan. Okay, because if I talk to you, if I explain one by one, 45 minutes will become 45 hours. Okay. Next. Walaupun saya tadi pompang kata, oh bagus natural product ni, semua okey, semua orang kata, semualah. So I love natural product, I love natural things. Tapi natural thing juga ada dia punya kelemahan. Bukan kelemahan lah, nothing's come with the optimal features. Dia mesti ada kekurangan dan kelebihan. Dan biasanya masalah dengan natural product ni adalah dia akan ada variability. Sebab itulah selalunya natural product ni susah diterima di dalam pharmaceutical market, sebab dia bila saya kata pasal pharmaceutical market ni, maksudnya sebagai sole treatment. Okay, macam insulin ke apa ke. Kadang-kadang kita tahu pokok ni mampu bantu kita dalam diabetes, dapat bantu elevate ataupun to treat the management for diabetes ke, hypertension ke. But the problem is sometimes untuk kita menaik taraf natural product ni adalah disebabkan oleh variability in quality. Sebab kadang-kadang kita ambil different vicinity of that plant sources. Kadang kita ambil dekat tanah negeri ni, walaupun pokok yang sama, ambil kat negara tu, tak sama dia punya isi dalam dia. The phytochemical dia tak sama. Okay, that is why it's hard to maintain. Compare dengan yang sintetik, because sintetik kita campur-campur dapat. Kan? Dan kita boleh control kat situ, dia punya structure mesti sama punya. All the time, from batch to batch. Tapi untuk natural product, itulah cabaran dia. Okay, it's hard to maintain the variability in quality. Okay, and then lengthy discovery process lah. Kadang-kadang sebab terlalu banyak option yang ada. Kita nak buat bioprospecting. Kita pergi kat hutan ni, ada berjuta-juta spesies biodiversity. Jadi nak trace satu-satu tu jadi ambil masa yang lama. Betul? Kan? And then scalability issue. Scalability issue ni pun antara benda yang kadang-kadang kita oversee lah. Kita tak berapa nak nampak. Okay, contoh, kalau kadang kita pergi, kita dah dapat identify. Okay, this pokok ni, okay, dia punya compound ni ada satu compound X fanata kan, contohlah kan. Oh, ni paling bagus ni untuk treat cancer. Okay, tapi masalah dia ialah pokok ni hanya wujud dekat Amazon, contoh. Okay, jadi macam mana nak cater satu dunia? Dan macam mana nak bagi kat satu dunia untuk dapat tu, and then dia memerlukan 10 kg pokok untuk dapat 1 miligram. This is the challenge in natural product research. Jadi walaupun challenge tu tak bermaksud dia mematahkan semangat kita. Betul? I don't care, I like this plant. Tapi saya nak overcome the challenge. Next. Okay, so how we can address this challenge? Yang tadi ada variability lah apalah. Okay, one of the way is kita standardize extraction technique. Okay, standardized extraction technique also can be done using quality by design. Okay, yang tadi Dr. Lazman ada share dengan kita, apa dia punya step-step yang kita perlu ambil, okay, untuk kita optimizekan the whole process, so that with the little resources, okay, kita boleh optimumkan supaya tidak membazir. Okay. Next, kita juga boleh guna high throughput screening. Tadi itu kita ada banyak sangat. Dia time consuming kan, macam nak trace one by one compound kan from the plant yang kita ada. Jadi kita boleh buat high throughput screening menggunakan in silico tools. Nanti kita tengok apa benda in silico tools ni. Okay, and then of course we do the interdisciplinary approach. Okay, by collaborating with industries, with chemists, dan various people from various fields. And then we also can integrate the technology that we have these days, okay, AI lah, whatever, okay, into the quality by design to enhance the discovery, development until the commercialization. Okay, so from natural, jadi apa kaitannya natural dengan computational ni? Okay, so how in silico tools can accelerate natural product research? Okay, in silico tools can bridge. Okay, dia akan menyambungkan, okay, two different knowledge, two different area by enabling the rapid identification and refinement of promising candidates. So please kita boleh guna this technology to help us back to accelerate the research. Okay, next. Okay, so biasanya kita boleh tengok lah kan, the process of drug discovery, mula-mula kita cari target lah, lepas tu kita buat in vitro, in vivo, toxicity dia, lepas tu kita buat clinical trials, barulah pergi review dan approval. Yang ni berkali-kali kamu akan nampak benda ni, the process, sampai dah hafal lah proses dia kan. Okay, next. Okay. Alright, kalau traditionally kita tengok ya, drug design ni boleh take up to 20 for a single drugs. Can take up to 20 years of development, starting from the identification. Siapa boleh tunggu sampai 20 tahun? Siapa boleh tunggu? Okay, masa kita COVID dulu, kan? Okay, masa kita COVID dulu, itulah dia kemuncak in silico works. Okay, how this in silico works help the humankind to find the solution ataupun vaksin daripada menggunakan teknik komputer. Okay, jadi mereka boleh speed up the process from the identification until the clinical, boleh shorten, instead of 20 years dia boleh shorten half, at least half, maybe less than 10 years juga boleh approve drug tu. Tapi untuk yang emergency case macam COVID yang dulu tu, okay, diorang terpaksa, FDA terpaksa approve cepatlah, okay, kalau tidak tak boleh nak solution. Tapi apa peranan in silico pada waktu itu adalah repurposing drugs. Okay, sebab kita dah ada available data of the approved drug yang ada sekarang ni, kita dah tahu structure dia, kita dah tahu dia punya interaction and everything. So kita try to match with the virus. Okay, dia mampu tak berinteraksi dengan virus. So we repurposing the drugs. We look at the interaction, we look at in the computer simulation, and based on that simulation, dia orang try run lah sikit for the preclinical, and they move forward for the clinical phase more faster than the traditional drug development process. Okay. Next. Okay, so what are the technology in computational in drug design yang ada sekarang ni? Okay, basically kita ada various techniques for in silico. Okay, one of it is molecular modeling. Okay, molecular modeling this include to generate. Okay, macam virus COVID tu. Okay, the emerge kan, dia orang isolate the virus. They study the DNA sequence. Okay, they study the DNA sequence. They extract the virus from the patient. Kan, dia keluarkan. They study the virus, and dia dah dapat dia punya protein sequence of that virus. So bila once dia dapat protein sequence itu metabolomics, genomics semua tu lah. Okay, sebab kita tahu nature of protein ni, okay, dia akan ada code. Dia ada code khas dia, this fragment until this fragment, dia akan hasilkan satu enzim ke apa-apalah, any compound. Okay, so dia adalah decoding. Jadi bila diorang dah understand the code of the virus, then from there they do the alignment sequencing, and then dia dah boleh tahu yang ini boleh dengan komputer lah kan. Kita boleh tahu dia punya folding dia supposedly macam mana and how to target that. Okay, dan kita boleh create satu drugs. Okay, kita tahu drugs tu setiap drug dia akan kenal dia punya target. Macam contoh kita bagi drug insulin. So insulin dia akan pergi kepada insulin receptor kan. Okay, so that is the target. Mana drug itu dia target ke insulin receptor. Jadi the same goes with the virus lah. So yang kita nak target untuk COVID tu, dia ada dia punya crown kan. Okay, crown dia tu saya lupa lah nama apa. Dia punya nama protein untuk crown tu. So dia try to find based on the structure of the crown. Okay, we try to develop drugs that can fit into the crown. Bila dia fit into that crown, dia akan inhibit the whole process of reproduction of that virus. So virus tu tidak akan reproduce, dia tidak akan membiak lah. The target. So the target, that is the drug target. Jadi kita can create a drug target based on that structure using computer. The same thing if we have the candidate from the natural resources. Okay, sebab orang suka natural kan, jadi we try to find from the natural. And then we do the protein modeling macam tadi kan saya mention lah. Based on the sequence, we can predict the protein. Do you know that actually if our DNA sequence, katalah species A dan species B, dia punya DNA sequence dia ada similarity of 70% and above. Okay, dia ada similarity, maknanya dia daripada sequence dia DNA yang pertama sampai ke 100, okay, 70% sama dengan species B, the protein folding will be the same. This is the homology study lah. Maknanya kita boleh kata, kalau kita nak generate, I think Dr. Laksana specialize in peptides. Okay, so peptide, kalau kita nak create peptides yang mempunyai structure yang lebih kurang dengan drug yang kita nak, so the sequence tu, this is actually special for enzyme drug lah, enzymatic drug. Okay, jadi kita boleh create the sequence at least 70% the same with the actual drug. Sebab kita tahu bila dia 70% dia akan folding the same way. So bila dia folding the same way, the structure will be the same and it can fit to the receptor. Sebab kita tahu enzim kan, dia receptor dengan lock and key kan. Dia ibarat dia macam kita ada satu, katalah peti harta karun. Peti harta karun dia ada mangga kan. Okay, kita nak break the treasure. Dia peti besi tu. Jadi kita tahu dah dia punya bentuk kunci tu sepatutnya macam ni. Okay, jadi kita try untuk cari struktur kunci yang boleh fit dan boleh buka kunci. Itulah konsep asas untuk drug designing ni. Okay, structure based structure-based drug design. Okay? And then kita ada juga teknik virtual screening. Okay, virtual screening ni dia macam kita buat screening dekat dalam lab lah. Kita test satu-satu, tapi kita buat dalam komputer dan dia tolong kita jimatkan chemicals apa semua sebab dia dah ada database dia. Okay, orang thanks to the older generation of researcher yang dah bagi kita data. Okay, data daripada data itulah kita masukkan dalam database dan bila orang lain nak access data tu, database tu, dia boleh trace down. Dah ada dah bentuk, orang dah kaji dah sebelum ni, dalam pokok ni dah ada compound A. Jadi kita tak payah ulang dah lah. Kan? Katalah dalam padi, kita tahu dalam padi ada compound A, B, C. Kamu nak ke pergi kutip padi, buat extraction apa semua? Tak payah dah sebab orang dah tolong check untuk kita. Kita just access the database. Okay, so from that database yang kita ada tu, kita gatherkan, and then daripada situ je kita boleh screen sebab kita tahu dah structure dia macam ni, kita try docking kan. Okay, kita try docking kan dalam komputer, dia boleh masuk tak dekat dalam kita punya peti besi tadi? Lock dia masuk tak? Tak masuk. Okay, bye-bye. Try lagi a second. No, bye-bye. So tak perlulah kita masuk dalam lab, buat lagi testing, try and error, buat kat tikus. Oh, last tak jadi lepas sebulan. Okay, so we do it in the computer. Thousands of compound can be screened using virtual screening within, depends on how your computer processing lah. Okay, so it can take around, kalau macam 5,000 compound 5,000 compound, kita target kat satu compound, sehari boleh siap. Sehari boleh siap, provided that your computer is okay lah. Kalau komputer punya RAM pun rendah, is quite hard. Okay, and then we do the drug repurposing macam yang saya dah mention tadi. Okay, next. Okay, so this is some of the tools again yang kita ada untuk computer aided drug design. Bila saya cakap pasal in silico means itu computer aided drug design. So kita ada molecular dynamics, we have structured drug design yang tadi saya cakap tu lah. Okay, yang try find the lock key tu, okay, and then ADME prediction. Okay, siapa pernah dengar ADME? Biasa dengar ni farmasi ni, kan? Apa dia? Absorption, distribution, metabolism, and excretion. This is the 4 main aspect of pharmacology yang kita kena tengok. Okay, biasanya kita boleh test dalam lab lah, tapi disebabkan teknologi yang ada sekarang, kita boleh predict without doing it in the lab. We do it in the computer. Okay, dia ada tools lah dalam web base. Kita ada Swiss ADME. Okay, katalah kita suka pokok tadi kan. Kamu keluar kejap lagi kan nampak ada pokok tepi jalan. Kutip. Lepas tu identify, ambil Google, ambil handphone, ambil gambar, Google tolong beritahu kita ini pokok apa. Okay, so nanti kita try tengok dalam database apa yang dah orang report tentang pokok ni. Okay, so from there we can do the ADME prediction. Oh, pokok ni toksik ni. Macam tu, contoh. So kita boleh tengok absorption dia apa semua based on kalau dia ada dalam database lah. Kalau tak ada orang kaji lagi, kamu jadilah pioneernya. Okay, kamu add up to the knowledge for the community. Okay, and then kita ada QMMM. Okay, QM ni quantum mechanics, molecular modeling, and then we can have we do also in silico works, protein structure prediction based on the protein sequence. Yang ni kena work with orang genomics. And then kita ada juga ligand based drug design. Okay, next. Okay, ini saya dah boleh ni belum? Okay, so ini contoh lah kita ada produk natural sources yang kita berminat ni. Okay, kita dah dapati dia ada compound mevastatin ni contoh. Okay, so kita boleh tengok macam mana dia punya pharmacomodel dan kita try dock. Okay, kalau kamu nampak dekat mana dia punya. Okay, so ini adalah compound dia. Okay, yang ini adalah protein yang kelabu ni. Okay, so kita try tengok interaction dia dalam binding pocket ni. Okay, kalau dia boleh fit in the binding pocket, means it has interaction. So that is how the in silico we can look at in silico. Dari dia adalah dia punya tools dia apa semua. We are not going to see it Kalau cerita ni tak habis kan? Okay, next. So ini some of in the database. Pokok-pokoknya lah. Saya pun tak ingat ada pokok apa ni. Sebab saya ambil pun daripada Google. Okay, next. Tak jelas. Tak jelas tak apalah. Nanti tengoklah dalam apa? Link Canva tu boleh tengok gambar cantik-cantik. Okay. So ini duk ulang-ulang benda yang sama. Okay, next. Next. Dia tak keluar gambar. Tak apa. Next. Pun tak keluar gambar. Dia nak suruh habis cepat tu. Okay. Tapi dah keluar dah. Okay, so how we integrate. Tapi kabur. Saya kabur ke kamu kabur? Kita berdua kabur. Okay, tak apa. Kita boleh tengok dalam Canva. Tengoklah, penat saya buat Canva tu tahu. Nanti balik boleh tengok. Oh, best lah. Best lah benda ni. Macam tu. Okay, next. Okay, so impact of in silico. So saya harap sampai ke tahap ini, it has opened up your mind that actually there is more advanced way in pharmaceutical development. It's not just like testing in the labs, but also we can enhance whatever technology we have today to accelerate the pharmaceutical research. Okay, so what in silico help us is by transforming the pharmaceutical research from the natural product, and together they can address the global health challenges with speed, precision and sustainability. Kenapa sustainability? Sebab with computational we can reduce the cost, the consumption of chemicals yang kita gunakan dalam lab untuk test. Kan? Kita dah jimat kat situ, kita jimat dan penggunaan chemical juga dapat dikurangkan sebab kita enhance, kita buat semua dalam komputer, tak perlu waste our resources, tak perlu waste our money, tak perlu waste our chemical untuk buat test. Komputer dah boleh tolong kita. Okay? So, we want to change. Okay, we want to change from random screening, okay, against disease into a rational drug design and testing and speed the screening process and do the efficient screening. De novo design. Ini pernah dengar tak de novo? Okay, de novo ni maksudnya new. Create new.
Ibu Ainun?
Yeah.
5 minutes left.
Okay. Towards the end. Alright. Okay, so we do the integration into design process and we can what we call eliminate the failed drug fast. Okay, next. This is about QBD, maybe because Dr. Laksana has helped me as well in explaining, so we can skip that. Okay? So what is QBD? Let us skip. Next. Okay, so ini je lah. Okay? QBD is actually the target, the prime aim of QBD is to achieve the right quality product by the right first time. Itulah functionnya. Okay, so next. 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 Next lah. Sampai ke... Okay, dah. Stop. Okay. So here, how important of QBD in herbal medicine, natural product development. We want to standardize. Ingat problem kita tadi apa? Kalau kita nak develop natural ni, is standardization because of the quality variability. And also kita nak make sure the process is optimized. Okay, that is why we need to implement, we need to insert the framework of QBD inside the development itself. Okay, and then to ensure that because QBD is a systematic and scientific way of proving that your development, so it can gain more trust and as well and helping you to get approval for commercialization. Okay, next. So I hope by now you can see the synergistically role of these three areas, which is the natural product, in silico and QBD. Okay, so the natural product give us the resources, then we use the in silico to enhance the resources, and with QBD we optimize the resources for drug development. Okay, so as the conclusion, by integrating natural product, in silico tools and QBD can offer a transformative solution to address the global health challenge. So this approach can accelerate drug discovery, enhance quality, that is important, and ensure sustainability. Okay, so for future, maybe we can do a collaboration across discipline, not only in the single discipline, across the discipline to unlock the future breakthrough. Okay, so let's unite the science and innovation to create a healthier, sustainable future. With all that, thank you for your attention.
Okay, thank you very much Ibu Ainun for the really nice presentation. And hopefully our student is not sleeping. They're really active. And how are you guys? Fine? Okay, we will continue with our last speaker. Are you ready? Ready, still, yeah? Okay, for our final speaker in this session, we have Prof. Sabir joining us online. Let me check him by online. Okay, is he joining already? Hello, Prof. Sabir, how are you? Waalaikumsalam warahmatullahi wabarakatuh. How are you today?
Very good, very good.
Very
I'm
good.
in Chennai.
You are in
morning.
Chennai. Yeah, we
Yeah.
actually met in Chennai, Prof. Sabir, right? Yeah,
Yes.
when we were had conference in Chennai, India. Yeah, we met in India last year on August, right? Or I forgot about the month. Yeah. Are you doing great today?
Very good, very good.
Very
Alhamdulillah.
good. Yeah, you ready with your presentation?
Yes, I have
Yeah.
shared it. Can you see it?
Okay. Wait, I have to read your curriculum vitae. Please wait a moment. Okay. Okay. I need to introduce our third speaker. He is Dr. Shabir Syed Abdul. Professor Dr. Shabir Syed Abdul. Unfortunately, today he is joining us online, but I hope someday we can invite him here together with us in our university. Okay, okay. Professor Sabil is currently a professor in a graduate institute of biomedical informatics, College of Medical Science and Technology, Taipei Medical University, Taipei, Taiwan. Previously, he is an adjunct professor at School of Gerontology, Health Management, College of Nursing, Taipei Medical University, Taipei, Taiwan, and joint appointment professor in School of Healthcare Administration, Artificial Intelligence and Digital Health Consultant, Principal Investigator at International Center for Health Information Technology, Editorial Board, Journal of Personalized Medicine. For his education, Prof. Shabir doing the medical doctor degree in St. Petersburg Medical Academy in Russia. And then for master degree, Master of Science, he got from telemedicine and health from the University of Tromsø, Norway. And for the Doctor of Philosophy, he got it from Health Informatics from National Yang Ming University in Taiwan, with the thesis title, A Novel Approach to Visualize and Represent Meaningful Information About the Disease-Wide Association in Human. Okay. Today, Professor Sabir will talk about the artificial intelligence and digital solution for mitigating global health challenges. Okay, can you back to Prof. Sabir? Okay, Prof. Sabir, our audio is right.
Assalamualaikum
Okay.
to everyone. Can you hear me?
Yes, we can hear you clearly. Can you hear us
Wonderful.
here?
Yes, very
Okay.
well. Thank you so much for inviting me. It's my pleasure to share my experiences. I was hearing to previous speaker, which was very good. And you see, there are a lot of combinations what we talk now. However, I want to make clear my talk will be more focused on global health challenges rather than pharmacy in particular. So it make a good sense. We are focusing on both challenges and pharmaceuticals, how to treat and make our world best place for us. Okay, so let's get started. I, okay, good. Before proceeding with my presentation, I want to shortly introduce Taipei Medical University. You guys might have heard about it. We have two campuses. Actually, it was established in 1960, almost say 60 years, more than 60 years. We have 11 colleges and 1,200 faculty and staff, over 6,000 students and 50,000 alumni, and there are so many students from Indonesia actually in our university. And we have 6 hospitals you can see there, where we give experience, real world experience for the students getting course in our university. And then comes my department, that is Graduate Institute of Biomedical Informatics. Our main research and application focus are biomedical informatics, precision health, big data analysis, health internet of things, telemedicine and mobile health, clinical decision support systems, artificial intelligence in medicine. And lately, I am more focused on virtual reality and digital twins in healthcare research. So anyone interested in any of this field are more than welcome to contact us, and let's see how we can proceed with your this thing. And then this is very important for us to understand, because our previous speaker also was talking about computer aided drug designing, all those things. But how this innovation comes, why we are talking about information and communication technology, because we have four innovations in healthcare industries. The first was the X-rays. Before X-rays, we used to treat the patient only by naked eyes, what we see outside. But with the X-rays, we get into the body, we can see the bones. It's not only the bones, we can see the fluid, we can see what's happening with the tissues. So that is how we diagnose different things. So X-ray changed the way we practice this. Then come antibiotics. If you remember, in World War One, a lot of people were dying with simple infection, wound injury, just because it got infected and there is no antibiotic to treat. But then comes antibiotic. Again, it changed the way we are practicing our healthcare. That is how it changed. The third innovation was integrating computers into diagnostic imaging, these things. Because if you see MRI, CT, what are these things? Yes, they combined different X-rays, especially CT scanning. MRI has different functions. It uses magnetic resonance to create an image by seeing the density of our tissues. But overall, the main purpose is to process the images and make it a one image, so that we understand the better, we understand the tissue density and do better diagnosis. Then comes the fourth innovation, that is information and communication technology. This brought us a broad Pandora of opportunities to process the data which we were unable to do with our like naked eyes or humanly. But now we have huge big data sources, data analytics, then comes AI, then comes LLMs, there comes so many different things. And that is the precise reason why we are focusing on computers, information technologies, and AI. Because you see, to get AI, the previous step was collecting data. If you remember, 5, 10 years from now, we were talking about cloud computing, we were talking about big data. Still, we were not talking about AI. But now we are talking about AI, we are talking about generative AI, we are talking about LLMs. So these are the advancements, right? So this is how it works. Then, as previous speaker, we need to focus on first the problem, then find the solution. What are the current problem of our healthcare systems or needs of our healthcare system? If you see that we currently focus on curative care, what it is, that means doctors are in hospitals waiting for a patient to go to them, express their complaints, symptoms, then the doctor will check the patient and start treatment. Unfortunately, this is not the right way of doing, because you cannot wait till the disease develops and many patients comes in late stages with complications. This all add to the cost, pain, sufferings of the patient and healthcare system, burden to the healthcare system. Therefore, since we have enough data available, we can do prediction, we can do prevention. So therefore, my first healthcare need, which we all, as a pharmacist, as a technician, as a doctor, as a consultant, as a surgeon, should focus is predictive care. That is preventive care. That is, without predicting, we cannot prevent. Because if I don't know what is the risk for you to get the disease, how I can prevent? If you start doing generalized prevention, like eat well, sleep well, don't smoke, don't take alcohol, all those things, people know it. Therefore, prediction is very important, because by giving your previous healthcare health data or disease information, your medication information, I can predict what is the risk of diseases for you, what are the risk for cancer A, what is the risk of cancer B. Then if someone has risk for some particular disease, it need not to be necessarily cancer, it can be liver problem, it can be kidney problem, it can be anything. Then precisely we can say, you have to do this, you should avoid this, so that he or she can at least prevent or prolong the onset of the disease. Prolonging onset of the disease means instead of getting cancer at age of 40, we are prolonging to age of 50. So we are giving 10 years of healthy life to that person, right? And then comes empowerment and engagement. This is also lacking right now, or at least from last till now. Why? Because we don't empower the patient. Only we ask, oh, okay, what's your problem? Okay, take this medicine, take 2 days, 3 days. That is not the way we need to do. We need to empower them by saying, okay, this is your disease. This was the cause. This is the reason. If you don't do this, it will come again. If you do this, you will not suffer again from it. So you need to engage them. You need to empower by giving more information, personalized information for that particular disease, and then engage them. And the third need is generalized to personalized medicine, because right now we are using all fit one for all. That means generalized. We use same medications for all patients, but same drug will not act equally to all persons because of our genetic information, because of our specialty in our genes, in our enzymes which are in our body. Therefore, you might have heard, for same drug, for one person it cures, for other persons it gives allergy. And for some persons, it makes such an allergy, even it is life-threatening allergy. Same drug. Why? Just because this person cannot take this medicine, right? Therefore, we need to go personalized medicine. Then comes long-term care for chronic disease management. Right now, what we do is that again, we wait for patient to come back, ask, how are you? Continue same treatment. Or if it is getting worse, increase the dose. So that is the way we are doing. If the patient drop, we have no idea what happened with the patient. So there is no long-term care of chronic diseases. If we involve all these things, what we added, prediction, prevention, empowerment, engagement, then we can do a much, much better way of long-term care and chronic conditions, stop or prevent complications of the disease and all those things. And if possible, obviously, we want to stop patients unnecessary visiting to the hospital. We can provide teleconsultations for patients at home to begin with. But if we feel necessary, we can, what you say, ask them to come to the hospital. And last but not the least is ensuring continuity of the care. As I told, if the patient don't come, we don't know at all who are not coming to us, who are not following up. Therefore, there should be a method by which we should ensure continuity of care. So I see some hands. So I don't know whether I should take the question now or we can put it after my presentation. Yeah, Dan, I need to take the question now or after my presentation?
After your presentation is okay.
Okay, so after
Yeah.
presentation.
Yeah, we will have a plenary
answer.
Q&A session together for 3 speakers here.
Perfect, perfect, perfect.
Okay, yeah.
So now comes one more introduction to whole healthcare, plus what I told previously, that is wearable device. Why wearable device? I will explain. Right now, we focus on electronic health record, medical records, which are in hospitals. We do analytics, we can do prediction, all those things. But if you add wearable device, what it happens? It gives real-time data, continuous data, what happens with the patients at home. That means physical activity data, heart rate data, so that we know how much the patient is physically active, when he is going to the bed, how long he is sleeping, all those information which directly empower him and engage him. Then these data also can be used for analysis to make clinical decision support. In that way, we can improve the quality of care we are giving to the patients. So this is something all hospitals and healthcare providers have to think how you can integrate the situation of the patient outside of hospital. Then I will talk a bit about data sets. From where we are getting data, what is health related data? Okay, what is health related data? Clinical health data is about how we born. 1 minute, I think something happening with my system. But anyway, 1 minute. Stop. Okay. So there are three components for any disease to occur. Number one, genotype, from how we born, from our parents, from our family, like ancestors. That components is already there, which make our genomic information, and that we cannot change. Okay. Can you see my screen, right? Okay.
Yeah, it's really clear. It's no problem
Okay, thank
here.
you, thank you.
Yeah.
Thank you. So, genomic information we cannot change because it is given to us by our parents. And if someone is prone to get diabetes, he or she have a high risk of getting it. No problem. Then comes environment, exposotypes or exposomes we call. That means the environment where we born. That also doesn't depend on us. We just born, we we have born at that particular place. No, we can't do any changes to that. What I mean by that is someone can born in industrial areas with polluted air, polluted water around them, XYZ, right? Can be. But some can born in very good area in the village where there is clean water, clean air, everything is good. It's possible. So that is also we cannot change. But imagine both have same gene of getting diabetes. Okay, then simple logic. The person with very clean air, good water quality may not get diabetes as soon as possible. But the third thing is behavior. That is the most important thing. That depend on the person himself. If the person doesn't take care of himself or herself, then the disease comes in 20 years, in 30 years. But if the person is active, if he doesn't take lot of sugars, if he is physically active, then he can prolong the same disease for longer period. So that is exactly what I mean to say. And phenotype means all the patient profile, what we save in hospitals, electronic health record. That is diagnosis, procedures undergone, medications, lab values. So all these data we can collect, are collecting right now. We can do analysis of this and we can do prediction. Next come another important factor which I was talking, that is exposome. What is this? It's all about where we are, how much radiation we are getting, air, water pollution, diet, drug, how much stress we have in our life because of family issues, because of social issues, because of job, XYZ. And what are the infections we are getting? Life, again, behavior and lifestyle. That is one part directly. Other part we need to also focus, it's not only about pharmaceuticals or the drugs we are taking, but also about cosmetics we are using. That is one type of chemicals which we are contaminating our body, right? Same thing, polluted food or adulterized products, oil, milk, butter, what not, and even simple water, what we think, what we are drinking. So we don't know what is exactly there, how good it is. So that is the point where we need to come up with a environment, ecosystem, which will control each and everything, right? Therefore, I want to give some challenges of global healthcare so that we focus on these challenges to make our life better in coming years. That is predictive analysis for disease outbreaks, or even it can be predictive analytics for chronic diseases. Also, we should focus, especially developing countries like India, like Indonesia, and our Vietnam, Thailand, how we can promote telemedicine and remote healthcare access, because still there are remote people in the periphery. It's not easy for them to get simple GE, like a general doctor's examination. Understand? Therefore, we need to go with digital health education and awareness, because now internet is everywhere, smartphones are everywhere. Then we can provide digital health content rather than just watching TikToks or some reels. We can promote them to watch some health related reels, health related TikToks, right? And also, in that way, we can put this health inequality in inclusions. That means why there is health inequality? Because there is no connection with all these things what I am talking for the people in the remote. When you provide all this information to them, then it becomes health equity and inclusion. Combating antibacterial resistance. I am sure in Indonesia also there is a lot of, and our experts from pharmacy definitely agree with me, there is a lot of antibacterial resistance issues. We call it superbug, right? There is big threat of superbugs already happening. It's not like will be going to happen. If we don't control antibiotic, we will be in big trouble. And this is the big challenge which we need to focus right now and see how we can control what you say prescription of antibiotics, how we can control whom to given, how to given. And last not the least, again, global collaboration and data sharing. Data sharing is very important, crucial subject where no hospital wants to share any data, but there is a need. We can de-identify data because we need data not for pinpointing, re-identifying the person, rather than helping the people to get care and preventing same thing happening with the next population or next group of people. Therefore, global collaboration and data sharing also play very important challenge in global health. So let's see how we can go ahead with all those things. Now, we heard a lot about AI, but now I want to bring the challenge. What is we are facing regarding AI adoption in healthcare? If you see, these are the publications. This is till 2020. In 2024, there are more than 12,000 publications published last year related to AI. But the problem is implementation. You see how much is the research and how much is the implementation. Why? Because me as a faculty, as a professor, I publish at least 4 to 5 different AI models for improving the healthcare. And then what I do, I keep that in computer or in my folder. That's it. I wait for another student to come up with a new problem, with a new data set, and we start working. Why? There is a huge problem of converting this knowledge into clinical practice because we need to go through FDA approval, lot of paperwork and legal issues, all those things where we don't have time and resource. In order to do that, we need to spin off the company, we need to start the company, we need to go all this legalization, then only we are allowed to implement. Therefore, I am focusing on how we can make this process easier so that what all research we do, we can clinically do practice and benefit the patient at the ground. And this is the approvals records, how it's happening. If you see in 2020, it's getting more, and radiology is low hanging fruit because it's nothing to deal with. It's image, see image, do diagnose, very simple, straightforward. Therefore, it has been recognized much easy and much faster. And I think this is very important slides for whoever interested in AI in healthcare, because there are so many different things which need to focus when we talk about AI in healthcare. What is progress, what are challenges, what are opportunities. Obviously you can focus on the opportunities, or if you are from what you say public health, if you want to work interested doing the challenges, then you can see which challenge you can take and work on that. For example, as I told, regulations, responsibilities. For example, if the software AI model gave wrong answer, who is the responsible, all those things, ethical data use, equity, biases, all those things. Or if you are interested in opportunities, then you can focus on how you can involve in AI in different things, non-image data source, image data source, because if you see progress, majorly medical image interpretation is at the leading. All these things are getting easier approval because it's very simple, analyze the image and finish with it. Okay. At the end, what I do is just I give a real world example of Taiwan, how we are focusing on preventive and prevention, prediction and prevention, which I started my talk about. Here, there is something called My Health Bank. If you are unfamiliar, you can just go and Google it, My Health Bank Taiwan, which provide access to your medical information. Anyone who visited Taiwanese hospital, because in Taiwan there is single payer hospital, all the data from any hospital you visit, dental hospital, public hospital, private hospital, even GP you visit, the data has to go to the one server. Therefore, this server has opened for the population, because everyone has their IC card, insurance card. You put that card into card reader, connect to your computer, and you can see your data, how many times you visited, what diagnosis you had, what lab values you had, everything you can see. That data was used, and they use machine learning models to predict two things: cancer and chronic kidney disease. So this is how it looks. And also you can add your blood pressure, your heart rate, and your body weight, what all you want to do in same file. So they made it as a personal health record for all population. And then they do prediction of two diseases, liver cancer and chronic kidney disease. And you cannot see data of any other patient or any other person, no way, unless until you get the card, you get their password, you know their username, all those things. So this shows what is your probability of getting liver cancer in 10 years. So if there is high probability, then the doctor stop giving medications which affect on your liver, and ask the patient not to consume alcohol, not to consume smoking, and also ask them to be physically active. In that way, they provide a preventive care. Obviously, it is up to the patient, he or she wants to follow it or not, but at least they are doing it. But there are some issues with this preventive medicine. That is, there is no visible target. For example, if I ask all of you, okay, do physical exercise 20 minutes every day, you will not get cancer X. Then all of you started doing. How much time you are doing? 1 week, 1 month, 3 months, 6 months? You are not getting disease. But now the question is, you are not getting disease, just you don't get ever, or you are not getting because of this exercise. So it is repetitive and slow. We cannot see the benefits unless until we know another group of people who are not doing exercise. Someone of them got the problem. Then you can see, oh, okay, in this group, because we are doing exercise, we have very less prevalence. That they don't follow any preventive care. That's why they have more prevalence. Then we can understand. And also, people don't understand the probability. I mean, common people, if I say the risk for you getting liver cancer is 0.1% or 11%, what does it mean? They don't understand. Then science can't produce reliable, useful predictions. That means we cannot predict these things. We need to just keep on following. Therefore, it has low market value. People don't invest, government don't invest in that, but it is very important. That is my bottom line. What I want to say is this: technology driven possibilities are rapidly growing and increasingly becoming available. The question is not which technology can be used, but what clinicians and patients need. So please focus on the need, on your problem, what you are facing, and then find the solution to that problem rather than going with AI, generative AI, chat GPT. That doesn't work. So first point out the problem. Okay. At the end, I want to give the take home messages, that is, focus on needs, not on technology. Consider technology as a tool, not as a solution. Focus on preventive and early detection. Don't still depend on curing the patient after they get the disease. Technology should be used to empower and engage patients. Right? By saying that, I once again thank all of you for your kind attention, and I am really waiting to take questions from you guys. Thank you so much.
Okay, thank you very much, Professor Sabir, for the really nice topic. It's about AI, right? Okay, now we will move to the Q&A session. I will invite once again Dr. Anna to come here with us, join us here. Okay, in this Q&A session, the committee, please help me to distribute the microphone. Okay, once again, in this Q&A session, we will open for the discussion with all the speakers here. Once again, we have Dr. Laksana, we have Ibu Ainun, and we have Professor Sabir in online. You can ask the question to all of the speakers here. Don't worry, you can use English and Indonesia, but we hope you will try with English first, okay? Yeah. Okay, maybe in the session, we will try to open question for 3 participants here. Okay, 1, 2, one more? Only 2? Okay. You can start your question and please tell us your name, the institution, and also the question should be addressed to which presenter. Yeah.
Thank you. Let me introduce myself. My name is Ulfiatul Fauza. I'm question for Ibu Ainun. How specific software in computer-aided drug design can accelerate the process of new drug development, especially in terms of effective prediction, and how to overcome the problem that arise and in the content of new drug discovery when our rational approaches preferred over traditional approach. Conversely, when our traditional approach more relevant. Thank you.
Okay, directly? Okay.
All right, thank you for the question. The question is very long, and I have to, but I captured the fragment of the question. So you are asking how specific is the computational tools? Okay, is the question have three segment of question, I think. Okay, the first one you want to know correct me if I'm wrong. Okay, you wanted to know whether how the computational tool are helping, how accurate the computational tool, right? Okay, the second is how the computational tool result can compare with the existing result available. Is it correct? Okay, and the third one, okay, can you repeat the question? The third one is?
Content of new drug discovery when a rational approach, conversely when our traditional approach more relevant.
Okay, so you wanted to know how you wanted to let's say if you have the traditional one and you want to how you want to ensure the traditional one with the modern available. About the structure, is it? Yes, is it correct? So there are three questions within your inquiries, right? Okay, so let's answer one by one. So the first one is, I forgot again the question. The first one is how accurate, right? Okay, actually you can browse. Okay, you can browse. There are various research that has already proven. Okay, that has already proven that the computational with the existing result is in line. We cannot say it's really 100% the same. Okay, but usually the in silico works derive from the available data. Okay, means the available data is someone already has discovered and they deposit that data into the databases, and that is when we use that database to research back again. So actually the data is valid data from is a valid data from what we say is already been proved by someone else. Okay, so actually we wanted to see whether we can use that data to answer our search. Okay, I am not sure I am answering your question or not, but I can feel your concern is how accurate the computational result with the actual result, right? Okay. Whatever you do in computational will not be proven until you do the in vivo and in vitro test. You still need to do the validation. Okay, that is the main thing. But the computational can help you accelerate that process. Instead of you try and error, okay, the first step is to find the candidate first. Okay, you find the most promising candidate from the database. Okay, you have thousands of candidates from the database. You find the single one that is the most promising based on your structure prediction, based on the interaction prediction, based on the ADME prediction that you have complete in the computer. Then that single compound that you are interested in and you decided that is the lead compound that you want to proceed, then only you do the in vivo and in vitro testing to validate that. Okay, the computation only accelerate the process of try and error of determining and finding the lead compound. Okay, did I answer your questions? Okay, thank you.
Okay, thank you so much. Mbak Ulfiatul ya, sudah cukup paham?
Sudah, Bu.
Sudah.
Thank you, Ibu Ainun.
Okay, thank you very much, Ibu Ainun. Okay, for the second question, we have the second. Okay.
Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. Before going to ask my questions, I say thank you for the moderator, for the three speakers, for the rector, and all participants that has given me time. Before going to my question, let me introduce myself. My name is Rizka Yuliana Saputri. I'm from Setiabudi University. Here I have general question for address to the three speakers. And I have a question for Dr. Laksana, and I have a question too for Ibu Ainun. Okay, for the first question, that is general questions. As we know here, almost the participants that come from the bachelor degrees. So my question here is there scholarship for master degree in UITM, Taipei Medical University, or Rangsit University? And if so, what are the requirements to obtain them? The second one is as our theme today, that is global health challenges in pharmaceutical research. And as we know here, almost the pharmaceutical research that using English language, right? Meanwhile, we are from different countries, like from the Malaysia that use Malaysian language, that we are from Indonesia use Indonesian language. So my question here, how to increase the English language of us so we can understand easier about the pharmaceutical research, like the journal that using English language. And here I have a question for Dr. Laksana from Tangsit University. My question is our topic, analytical quality by design in pharmaceutical development. So my question here is, what are the biggest challenges faced by pharmaceutical industry in implementing AQBD or analytical quality by design? The second one is that you have to talk in us, or you have explained us about comparison between QBT and QBD approach. So in number 2, that explain in QBT process development is empirical univariate experiments, and for QBD that impose systematic process development multivariate experiments. So my question here is, can you give me the example of the univariate experiments and the multivariate experiments? And the last question is for Ibu Ainun. My question here is that we have talked about in silico, right? My question here is, what are the limitations of using in silico tools in pharmaceutical research? Okay, thank you very much. I'm so sorry if I take a long time and really more of questions. I say thank you very much for the attention. And the last I say, wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much for the question. What is your name? I'm sorry once again. Rizka Yuliana Saputri. Okay, I will note your name. Okay, maybe this is really, maybe our students want to know, yeah, about scholarship. Yeah, yeah. Actually, I also want to know if I have any more scholarship. If I can go, I will also go there. Okay, we will start with Dr. Laksana first. Yeah, please answer the question.
Okay. The first question about the graduate program, right? At Rangsit University College of Pharmacy, we offer the graduate program in master degree and in doctoral degree. You can go to the website, search for Rangsit University first, and then go to the College of Pharmacy. We can arrange the course in research base and can talk in English. And the second question about the challenge of analytical quality by design and quality by design in the pharmaceutical industry, right? As you know that when we need to do the quality by design, we need the software. And the computer software is need the understanding and maybe need the training that you need to input the parameter in the software, and then it will decide. It depends on the target that you want to explore, and then we need to run the experiment, run the test, and collect the data, and then input back to the computer. So it will generate the response, and we will see the response, how we change those parameters, how it affects the output. Then we get the optimize. So in the industry, maybe should collaborate with the university or the research lab, and then get the person who will be the key responsible. Some people said it may take time and maybe cost some money. Actually, at the beginning, I think it's like the investment. But if you do it at the first time, and then you get it and get the method for the routine, it will be benefit for you, that you can get that. And when you change, you can get these data for the analytical method development, and you can have your data when you want to submit to the registration agency, it probably is in the long term, in the long run. So just need to difficult at the first time to start. And you want to the example of univariate and multivariate analysis, right? It's like the computational and mathematics and statistics. For the univariate, I think univariate is one. For example, that I give the example of the analytical method development. If you want to start with the one stationary phase or column, so you need to decide that you're going to use mobile phase, one mobile phase or two types of mobile phase and ratio or not. You need to change one factor. For example, you change the ratio one time, run the HPLC chromatogram and see, and then if it not get the peak result, you try another condition. But for multivariate analysis, you have the factor like the ratio, pH, flow rate, and then you input that parameter in the computational design. Then you have to define your response that you want to see. It's going to be the resolution of the peak, or it will be the theoretical plate or peak symmetry. And then you will see which factor will affect your response. This is multivariate, and you can do at the same time or simultaneously and summarize or categorize your result, your response. And I didn't remember the question. About the language, or you mean increase English language? Yeah. Actually, in Thailand, English is not the first language for me too. So basically, I learned in the school. And if you like to listen to music or watch the movie to practice the hearing and speaking, and don't worry the grammar. Try to speak. Then we can communicate and understanding. But for scientific research, probably need to read a lot of the article in English.
Okay, thank you very much, Dr. Laksana. Please, Ibu Ainun.
Okay, Assalamualaikum, Rika. Is it Rizka? Rizka. Okay, thank you for your question. Do I need to answer again the question, the first and second question about the postgraduates, the postgraduate in UITM?
Of course, Miss, because you're different university, right?
All right.
Okay, thank you.
All right, so the first question is about our postgraduate studies. Okay, so in UITM, specifically for pharmacy, faculty of pharmacy, we do have two types of masters. Okay, we have master by taught course. Taught course means you go to the class. Okay, they do the class lah, you took the class, exam, and everything. And the second type of masters is master by research. Master by research means you do research for 2 years. Okay, and you complete your research and you need to pass your viva. So the examination, you only have one examination, that is the viva lah. So you do the research and you have, okay, the difference between taught course, we call it taught course, and also by research. Okay, the taught course, you have 2 semesters. So you have around, I'm not sure how many credits, but there are few subjects that you need to pass. Macam biasa lah, okay, macam biasa, you macam degree jugalah. Okay, but in 1 year. Okay, within 1 year. So by the end of 1 year, you already got your masters lah. Okay, itu kelebihan dia cepat, 1 tahun. Okay, tapi yang kaedah kedua, iaitu master by research, usually you will be appointed with one supervisor. So the supervisor is your dosen. So the supervisor will supervise your project. So you do the lab works, you do your field works, lab works, or whatever that is in the context of your research. Okay, and you must complete the research within 2 years. Okay, but you will gain a lot of new knowledge for that. That is the, if you wanted to know which one is suited the most, is actually you need to know what is your objective of pursuing postgraduate. Are you pursuing your postgraduate because you like research, or you want to proceed your postgraduate because you want the qualification? You need to ask yourself. So if you just wanted the qualification, like if you want to be a docent in academy, okay, that require you to have a master degree, then you can simply go for the third course to jimat masa lah, to hemat kita panggil dalam bahasa Indonesia, hemat, to shorten your time. Okay, but if you have the patience of continuing to PhD and you love research more than anything else, okay, then maybe you can consider by research, because by research you can expand not only for the context of your study, but from research you can gain more collaboration, more insight of the current situation, okay, and will help you to understand what can you give to the community back. Okay, instead of you just go to the class, passing your exams, but the research field is much more open, and it will polish your skill better. It is my opinion. Okay, but in UITM we have these two type of master, depending on your objective of pursuing postgraduate.
How about the scholarship, Miss?
The scholarship, okay. Yeah. For UITM, okay, the faculty itself, we don't have scholarship, but usually the supervisor, when you, if this is only for research, this is for only, I told we have two types, right? This is only applicable for research students. Okay, for research student who wants to proceed with the postgraduate master in UITM, if your supervisor have grants, research grant, they can biaya your tuition fee, your life allowance, monthly allowance. Okay, in Malaysia for master is 2,300, which is equal to 1,000 ringgit is equal to 3.5 juta. 3.5 juta. Around. 1,000 Malaysia is 3.5 juta. So if 2,000, 7 juta. 7 to 8 million per month. If you do by master. Coursework, by research. But if you do for coursework, there will be a tuition fee, because we need to pay the credits. So that one don't have beasiswa lah, only for the research. But if if the supervisor has grants lah, usually supervisor who has grant only looking for students, because they need to pay the student within 2 years. Usually like that in Malaysia punya scenario lah. No specific scholarship for postgraduates. The third question is, what is the challenge, right? Sorry, do you have any question regarding the?
The second one is about how to solve or how to increase English language. Meanwhile, we are different countries like Melayu.
Okay, I hate to reveal this to you, but Rome does not build within a night, okay? So there is no shortcut in knowledge. You cannot suddenly become fluent in something or have very much knowledge in something without seeking and practicing. Okay, I hate to say that there is no shortcut. So means you need to read more and practice more in communication and read journals as much as you can. Okay, because if you started to read journals, you will see the style of language is almost similar. So from time to time, you will start to figure out and you can predict what is going to be said in that journal actually. So there is no shortcut. You need to read more, you need to practice more. Okay. Also in Malaysia, English is not our is not our first language as well. Okay, so we need to practice. Okay. Is it okay? Okay. The third question.
The third question about in silico, that is, what are the limitations of using in silico tools in pharmaceutical research?
Okay, the only, let's say the main challenge for in silico works is that your result will not be dim. Your result will not be dim full until you do the validation in vivo and in silico. Okay, we must see that the in silico works is only to help you to accelerate the first stage of drug designing. Okay, in any way, you still need to validate in vivo and in vitro. That is why in research for pharmaceutical, you cannot go alone. Means that if you are very good at computational, you cannot just do that. You have to collaborate with people who are good in vivo and also good in vitro. Then only the whole discovery is holistic. If not, it's just a fragmented. So the only challenge is that your result is not deemed validated until you do the in vivo and in vitro. Okay. Am I answering your question?
Of course, Miss. But I have a question too about what is advantages or disadvantages in silico, Miss?
Apparatus?
Advantages, manfaat.
Advantages.
Yeah.
Okay. Of course, like any other research, okay, you need to have tools, right? Like if you do in vivo, you need to have skill in pipetting. You need to have skills in analyzing the HPLC and whatever. The same goes for in silico. You can run the simulation, but did you really understand the output of that simulation? That is the tools or the skills that you need to develop. Okay, I mean that is the skill that need to have for a computational chemist. Okay, so you need to understand the basic of your algorithm, and also that is why computational is something like is a hybrid of mathematician, statistician, bioinformatics. Okay, so people with bio knowledge and people with the chemistry knowledge, biochemistry and computational need to combine. Am I answering your question?
Of course, Miss.
Okay.
Thank you very much for the explanation. Okay, thank you very much. Really wonderful discussion. I think you are also interested in silico? A little bit, okay. Yeah, maybe Pak Dekan, after this meeting, we need to facilitate students. Maybe we will make like one day English session like that, Pak Dekan. Okay, Mr. Professor Sabir, are you still with us?
Yes, I am with you guys.
Yeah, okay. This is the question from our participants. She wants to know about, is there any scholarship in your university, and probably you can introduce a bit how to apply and how is the chance to go to the master degree there or maybe PhD? Okay.
Yes, thank you so much. As previous speaker told, our university, Taipei Medical University, also provide masters and PhD, and we have College of Pharmacy, and especially with the emerging technologies like what you say subjects like precision medicine, pharmacogenomics, pharmacovigilance. There is a lot of scopes for you guys to come into, and now you don't need to develop depend only on developing a new molecule or discovery. You can also focus on repurposing of the existing medications, which is also a big field emerging. And coming back to the scholarship, yes, PMU provide scholarship, various scholarship depending upon your background, your knowledge, your interest, which will be scrutinized by interviews, by the certificates you are submitting. So there are a lot of opportunities for scholarship as well as for learning, get training into it. Anything else you want from my side?
About how to solve the problem, like we really decrease in English language, like how
So
to
again,
increase our English language
see,
meanwhile to know
I
about
do understand the
pharmaceutical research
question, but
by using English language. Thank you.
two speakers already thought it's only by practice. You cannot learn in one day or one night. So you just keep practicing, reading articles, doing things, and taking the courses. Now you have so many MOOCs, massive open online courses. You have videos instead of watching movies. You watch English movies or English content. That gives a lot of knowledge. Really, it makes the world easier. So just go ahead and listen, speak, and then you will get experience into that.
Okay, thank you for the explanation.
Okay, thank you very much, Professor Sabir. Professor Sabir, may we know, is there any faculty of pharmacy in your university or any related medical science in the
We do
university?
have, as I told, we have complete College of Pharmacy with the different department branches into that. Plus, I wanted to emphasize we also have Global Health Department, which is
I
totally
see.
different.
Yeah.
So people interested in global health issues can go there. People interested in precisely pharmacy can get into College of Pharmacy. Plus, people interested in computations, AI, bioinformatics can come into my department, that is biomedical informatics, where we again have lot of medications is analyzed. We do risk of diseases depending upon the medications the patients have been taken. You see, so there is lot of opportunities.
Okay. So, but unfortunately, we don't have the MOU yet between our university and your university. So, if possible, next time we should make an official MOU between our university. Is it
Definitely,
possible?
that is,
Oh, yeah.
yes, it is possible. Just
Yeah.
email and I will connect to our OGE, Global Engagement Office. Then you can go ahead from there.
Okay, yeah, that's
No
good.
issue.
So we hope that our student can get more opportunities globally, and probably we can visit in your university in the next future.
Most
Okay.
welcome. And there are so many Indonesian students, as I told, in
Oh,
TMU.
really? Yeah,
So
yeah.
many, yes, yes. Yes.
And um, in I think it's really it's really hot topic recently, right? In the medical science, in
It is.
developing really rapidly right now.
True.
Yeah. Now we are using many AI here. Okay. We will also say hello to the participant in the online session, which in the Zoom or YouTube. If you have any question, please, you can raise your hand and we can open the access of the microphone for you. And also you can type the question in the Q&A box. Yeah, our technician will help you to read the question. Is there any question in the chat box, Mr. Randy? Please check it. Yeah. Or in the YouTube probably. Please help me to check them. Okay? Meanwhile, we are waiting for the online, maybe they will also have question. We will open for the next discussion. Is there any question from the participant here joining us offline? Yeah, please remember, we will have special prize here. So we provide already many, I think good. Yeah, okay. We have one question there, and okay, with the, please pass the microphone to, stand up please. Microphone. Your number second. Okay. Where is the microphone? Oh. Oh, that one first. So 1, 2, and 3. Yeah? Okay, you can sit down first. Okay, the first question, please.
Okay, thank you for the opportunity given to me to ask. Let me introduce myself. My name is Laila Tudarisa Delianti from University of Setiabudi. I want to ask with Mr. Shabir Sheikh, what are the key ways that artificial intelligence and digital solution can address global health challenges, particularly in improving healthcare access, efficiency, and outcomes for underserved population? Thank you. That's all for my question. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Thank you.
Miss, yeah.
Thank you
Prof.
for
Sabir,
the
can you
question.
get it?
Yes.
Yeah,
I
okay.
listen the question. Yeah, it's very good question. So it's all about accessibility or reaching the people underserved population. As I told in my presentation, we can use technology to solve that problem by providing teleconsultation, by providing empowerment for the patients, engagement to the patients by connecting using mobile phones. So now you can use mobile phones to connect them, to send them the appropriate, tailored, customized information that the patient needs. In that case, we will be engaging them and empowering them. I think this is the one of the way to solve the problem of accessibility, so that the patients are in touch and they know what is happening with them and they can visit whenever it's needed, not like just travel so long and say, okay, everything is good or repeat same treatment. Those things should not happen. So once my short answer is yes, use technology, use telemedicine tools, use digital health tools in order to empower and engage the patients.
Okay, thank you very much, Prof. Sabir. Is it enough, Mbak Lailatul? Have any more question?
Enough. Thank you,
enough.
Mr.
Okay, thank you very much. Okay, because our time is limited, we will directly move forward to the second participant who want to ask the back one first. Okay, you got the microphone already? Not yet. Okay, you're number 3 then. I'm sorry. Okay.
Sorry. Excuse me for permission to ask for Mr. Prof. Dr. Shabin. Let me introduce myself. My name is Nadine. I'm from Setiabudi University of Surakarta. How get rate of COVID continues to grow throughout the world? Bagaimana cara menghilangkan COVID yang terus berkembang di dunia?
Bagaimana cara? Mikrofonnya dideketin aja, Mbak, nggak usah malu, iya.
Cara menghilangkan COVID terus berkembang di seluruh dunia.
Oke. Ada pertanyaan lagi? Cukup satu?
Satu aja.
Cukup, oke. Terima kasih Mbak Nadine. Thank you very much for Miss Nadine. Okay, Prof. Sabir, so the question is for you. Hello? Yeah. So,
Yes, I can listen.
Okay, she is concerned about the COVID. Yeah, COVID is still developing nowadays. It's not stoppable probably. So maybe with the AI technology right now, how can we possibly stop this COVID? Okay, that's her question. Please
Okay,
answer. Yeah.
again, true, very true, but as I told in my talk, technology is not solution, right? Technology is just a tool. Number one, COVID is actually old chapter. Now we learn everything about the COVID, symptoms, treatment, all those things. So the best way to prevent COVID, we know, is prevention, right? We cannot bring AI to prevent yourself. You should prevent yourself by using mask, by cleaning hand, by not getting into the people where there are so many people without mask, X, Y, Z. These are the preventive measures. So you need to follow. AI cannot help in this. Yes, AI has done its task by developing the vaccination in no time. What all our previous speaker was talking, computational drug designing, everything was done by using AI and computation to develop the vaccines of the COVID. Now it's up to us how we are preventing ourselves. It's not about only COVID, it's about all infectious diseases. So if you use mask, you are prevented by various lung diseases which gets into your body by your respiratory systems, right? If you clean your hands, you will prevented by many tuberculosis and salmonella, so many different type of infectious diseases. So it's all about your practice, lifestyle, how you are behaving, eat immune like good food, protein, so that your immune power is strengthened. That's how it works.
Oke, thank you. Mbak Nadine bisa menangkap? Bisa? Perlu diterjemahkan? Oh, enggak usah. Oke, thank you. Oke, we will move to the last question. Please, your question. Belum dapat mic. Mbak yang di belakang itu, masnya yang berdiri dekat pintu. Oke.
Okay, thank you for giving me the time to ask. Let me introduce myself. My name is Guru Resi Givari. I'm from Setiabudi University. I want to ask about what if singularity is under 70%, whether it can still make vaccination using computation? And can I help to find a solution? It's
Ditujukan ke siapa, Mas?
Miss Aini.
Ainun. Ya, Ibu Ainun. It's your turn to answer.
Sorry, Miss.
Miss.
Thank you. Okay, thank you for the question, but can you repeat the first one? I've lost the question.
What if the singularity is under 70%, whether it can still make vaccination using a computation? And can artificial intelligence help to find a solution?
Is it referring to the slide that I said 70%?
No. singularity make a vaccine.
Oh, okay. All right. Okay, so, okay, thank you. You can sit down.
Thank you.
All right. Actually, what your name is? Your name?
Guru.
Sorry.
Guru.
Guru, okay. So Guru is asking about the similarity. This is about the protein sequence alignment, right? Okay, so actually in my sharing session, I've mentioned something about protein sequence alignment. Okay, actually that technique is being used to predict protein homology. Okay, protein homology. Okay, like I give you example during the COVID. Okay, during the COVID, okay, remember that they say when the patient tu dia kita buat test dekat patient and then dia dapati kita dapat dia punya virus kan. And then we do the sequencing of that virus and we have the protein sequence of that virus. Tapi masa tu tak tahu virus tu datang daripada mana. Kan? Okay, jadi what the immunology punya, bukan immunology, apa kita panggil, the person who informatics, okay, bioinformatics, okay, diorang detect the sequence sebab nak tahu daripada mana datangnya virus ni. Daripada sequence tu didapati the DNA of that virus is similar to kelawar. Kan, ingat tak yang masa COVID tu, diorang kata virus tu datang daripada kelawar. So that is macam itulah diorang tahu mana datang benda tu. So that protein alignment sequencing tu is a technique can be used to predict. Let's say kata kita next time kita nak produce satu protein, kita nak synthesize satu protein yang dia punya, katalah protein tu daripada porcine, babi kan. So babi for Muslim kita tak boleh guna, betul? Tapi kita boleh synthesize based on the sequence. Okay, so kita boleh generate the sequence up to 70%, kena similar more than 70%, then only that protein can be folded the similar as the original porcine. From the swine resources. So dia dah boleh jadi halal lah sebab dia tak exactly from swine. Kita synthesize that protein based on the protein sequence. So that is one of the tool. Am I answering your question? Lagi question tadi lagi satu apa?
The second question is, can artificial intelligence help to find the solution?
Okay, for the AI, because AI we have different type of tools. So depending on your objective, what are you want to solve? What do you want to solve? Do you want to see the interaction? Then you can use the molecular modeling. You can do molecular dynamic simulation. If you wanted to design the AI tool is various tools. Okay, so of course it can help us, but it is not the sole solution. You still need to validate whatever the AI help us with in vitro and as well as in vitro. Okay, I hope I answer your question.
Thank you, Miss.
Okay, thank you very much, Ibu Ainun for the answer. Okay, ladies and gentlemen, because our time is limited, we have to close this plenary session. Yeah, once again, I'm very thank you for the speakers here, Dr. Laksana, Ibu Ainun, Prof. Sabir, and all of the participants, both here in person and joining us by online. Thank you for the active engagement, thoughtful question, and shared enthusiasm that has been instrumental in making this session successfully. Next, as we move forward,
we please give the big applause for the all speaker and also of the questioner. Okay, to close this session, thank you again for your participation and contribution. Now I will move hand time back to the Master of Ceremony to continue the program. Thank you very much.
Yeah. Thank you, Dr. Thank you, Apoteker Dian.
Ladies and gentlemen, the great material by our great speaker has ended. And now it is the certificate submission by Dean of the Faculty of Pharmacy Universitas Setiabudi, Dr. Apoteker Iswandi, Magister Farmasi.
Hadirin sekalian, materi hebat dari 3 pemateri hebat kita telah usai. Dan tibalah saatnya penyerahan sertifikat oleh Dekan Fakultas Farmasi Universitas Setiabudi Surakarta kepada Dr. Apoteker Iswandi, Magister Farmasi. Kami persilahkan untuk mempersiapkan diri.
And to our speaker, we also welcome to move on stage. Okay, big applause to the speaker today.
And after this, we also invite Dr. Apoteker Iswandi to symbolically present a certificate online to Professor Dr. Sabir Syed Abdul, MD, MSc, PhD. Sekali lagi kami ucapkan terima kasih kepada Dr. Apoteker Iswandi, Magister Farmasi yang telah berkenan untuk menyerahkan sertifikat kepada pembicara kita dan juga moderator kita. And now we invite to our speakers to leave the ballroom. teman-teman sekalian dan hadirin sekalian, kita sudah ada di penghujung acara inti, tapi sebelum itu nanti kita akan ada pembicara terakhir.
Dari Malaysia juga tentunya.
Tapi sebelum kita mau beranjak ke sesi berikutnya itu makan siang, kita akan mengumumkan 5 peserta tercepat untuk hadir pada hari ini.
Iya.
Apresiasi ini diberikan kepada 5 peserta yang hadir terlebih dahulu hari ini.
Kira-kira siapa nih?
Kira-kira siapa? Tadi yang merasa datang duluan siapa?
Wah, langsung angkat tangan semuanya ya?
Iya, langsung banyak nih. Langsung
Iya.
saja. Untuk yang datang tercepat pertama, selamat kepada Saudari Andini Prabandaru Siwi. Kami persilakan
Ya.
untuk langsung maju ke depan saja.
Boleh untuk langsung ke depan untuk nama-nama
Gak
yang
ada
telah
salahnya
disebutkan.
ya kita untuk datang cepet-cepet.
Oh iya, nggak ada
Datang dan
ruginya.
gasi itu nggak ada ruginya gitu.
Iya.
Langsung saja.
Oke, untuk yang kedua, Sepdita Indri Pramudianti. Oke.
Ini kayaknya satu geng ya.
Kayaknya satu geng.
Berikutnya ada Nurfiana Rahmawati.
Dan juga ada Vina Aditya.
Wah ini satu geng beneran. Dan selanjutnya adalah Nabila Valentina.
Oke, beri tepuk tangannya dong untuk
Ini adalah geng yang positif.
ya peserta-peserta yang paling disiplin.
Oke, berikutnya untuk penyerahan apresiasi akan diserahkan oleh Ibu Leli selaku si acara dari acara kita hari ini.
Oke.
Kepada Ibu Leli kami
mungkin
persiapkan.
Bisa lebih ke tengah lagi.
Iya, bisa lebih ke tengah lagi. Inilah wujud nyata bahwa mahasiswa USB itu selalu on time ya teman-teman.
Ya, dan hadiahnya disponsori juga oleh OMG.
Ya, dan setelah ini nanti kita akan ada makan siang. Oh iya, baik. Sembari berfoto, untuk teman-teman sekalian bagi penanya, nanti di akhir acara akan kami serahkan hadiah.
Nah, untuk itu jangan pulang dulu ya teman-teman habis ini.
Karena hadiahnya sungguh-sungguh menarik.
Iya, peserta
Dan-
teraktif
Iya.
akan juga mendapatkan hadiah yang tentunya hadiah ini bukan kaleng-kaleng.
Nilainya JT-JT-an.
Ya beneran
Totalnya. Dan langsung saja, masih
Oh,
mau foto
masih
ya?
mau bikin JJ nanti kayaknya ini.
POV datang ke seminar internasional atau
Iya.
apa lagi ini JJ-nya? Banyak sekali ini. Oke, nanti juga bisa tag OMG ya mungkin ya.
Iya, bisa minta tolong untuk tag OMG.
Oke, ladies and gentlemen, now it's time for us to take a lunch break. Dan untuk lunch breaknya hingga pukul 1 siang ya.
Ya, jadi nanti akan dikasih kesempatan untuk beristirahat dan beribadah bagi yang menjalankan. Tapi nanti pukul 13 waktu Indonesia Barat dihimbau untuk segera merapat ke ballroom.
Dan untuk pembagian tempat makan siang untuk sebelah sisi kanan dari
Ya.
kamera yang ada di tengah, sebelah sini, itu nanti akan makan siang di
Restoran.
restoran yang tepat ada di
Tepatnya ada di tadi pas jalan ke sini sudah lihat restorannya?
Ya,
nah, di
di
situ
situ.
tempat makan kalian nanti.
Dan untuk sisi berikutnya akan makan siang di
Dari stall 4, 5, dan 6 nanti akan makan siang di depan meeting room. Jadi di depan meeting room di Pamiluto, Kawung, dan Meru. Sudah tahu ruangannya? Nah, nanti di situ akan ada juga mini restoran dan untuk stall yang saya sebutkan tadi bisa makan siang di situ.
Baik. Oh ya, untuk
Ya,
tadi
di depan
lokasi
resto.
yang ada di sebelah sini tadi tepatnya ada di depan resto. Langsung saja nanti ke depan resto, nanti ada panitia yang mengarahkan.
Baik, untuk biar nanti jalannya tidak sumpek-sumpekan nih, Yemima, mungkin dari sebelah kiri saya bisa terlebih
Lalu
dahulu.
keluar, monggo kami
Ya.
persilakan. Dan untuk musola itu ada di basement dan juga di,
Di
lantai 10.
Kamar mandi ada di lantai 2. Keluar dari ballroom belok kiri saja, belok kanan, sorry, nanti lurus terus ada toilet laki-laki dan perempuan di situ.
Baik, untuk teman-teman sekalian dan hadirin sekalian, terima kasih karena sudah berpartisipasi secara aktif dalam sesi pertama. Dan hari ini teman-teman dan hadirin sekalian kami undang untuk bisa beristirahat, take a break,
Ya,
untuk
dan makan siang.
Kira-kira apa ya makan siangnya ya?
Wah, pasti kalau restorannya enak-enak sih.
Iya, nanti juga ya. Oh iya.
Oh iya, ada informasi tambahan untuk makan siang bisa dibawa ke ballroom juga. Atau bisa dimakan di restoran juga boleh. Sama di meeting room juga boleh.
Ya. Mari kami undang untuk yang sebelah kanan dari kamera bisa terlebih dahulu untuk keluar. Nanti setelah itu akan kami kode untuk sebelah kiri kamera boleh keluar.
Ya, dan kami ingatkan lagi jangan langsung pulang ya karena masih ada hadiah banyak sekali di belakang acara nanti tentunya.
Iya,
Yumi,
tentu
Pak.
saja. Nanti kita akan ada sesi berikutnya dengan Dr. Apoteker Rina.
Ya, betul
Dan
sekali.
nanti mungkin teman-teman bisa mempersiapkan pertanyaannya dan lebih aktif lagi. Karena
hadiahnya pada hari ini sangat luar biasa sekali ya.
wow, luar biasa sekali.
Ya.
Nah, ini seperti contohnya kakak-kakaknya langsung
Ya,
update ya, Kak.
update story ya.
Mantap. Berangkat seminar nggak bawa apa-apa, nggak skincarean, pulang bawa skincare.
Iya, borong skincare.
Mantap. Langsung satu geng glowing bersama.
Iya.
Grow up bersama.
Oke, nampaknya sebelah kiri saya sudah terlihat sepi dan di sebelah kanan saya
Boleh langsung
untuk
keluar.
segera bersiap-siap meninggalkan ballroom.
Dan kami himbau untuk tidak meninggalkan barang-barang berharga teman-teman ada di
Ya.
sini. Karena kami juga tidak tahu, mungkin ada hal yang perlu diperhatikan.
Ya, dan kami ulangi lagi untuk musola ada di lantai 10 dan di basement. Kamar mandi ada di lantai 2, tepatnya ada di depan restoran. Dan belok kanan lurus saja nanti akan terdapat toilet laki-laki dan perempuan.
Betul sekali.
Bagi peserta seminar yang belum registrasi ulang, diharapkan untuk menemui kesekretariatan di depan ballroom untuk registrasi ulang kembali. Sekali lagi, yang peserta seminar yang belum registrasi ulang dapat menemui kesekretariatan di depan ballroom.
Bagi teman-teman yang masih ada di luar bisa segera masuk karena acara berikutnya akan segera kami mulai. Baik, kami himbau kepada teman-teman dan hadirin sekalian yang masih ada di luar dari ballroom bisa segera masuk karena acara berikutnya dari materi terakhir akan segera dimulai. Baik, untuk teman-teman yang masih ada di luar, dimohon untuk segera masuk karena acara akan segera kita mulai.
Dan untuk peserta seminar yang belum registrasi ulang dapat menghubungi kesekretariatan yang ada di depan ballroom.
Kepada seluruh hadirin, acara hari ini tidak dapat terselenggara dengan baik tanpa adanya dukungan dari individu maupun organisasi.
Ya.
Dan untuk itu kami akan bacakan beberapa sponsor kita pada hari ini. Yang pertama ada PT Aneka Kurnia Utama,
Dan juga ada PT Tisan Nugraha Globalindo,
PT Putra Bengawan Sukses,
PT Farkositra Internasional yang memproduksi Oh My Glam,
PT Ditek Jaya Analytical dan Measuring Instrument.
Baik.
Baik teman-teman sekalian, mohon untuk segera duduk kembali karena acara akan segera kita mulai. Langsung saja, teman-teman langsung saja menempati tempat duduknya semula. Bagi teman-teman yang merasa bahwa sebelahnya belum hadir dan dirasa kenal, mungkin bisa dihubungi juga secara pribadi agar segera masuk ke dalam ruangan ini.
Kami infokan sekali lagi bagi peserta seminar yang belum registrasi ulang dapat menghubungi pihak kesekretariatan di depan ballroom untuk registrasi ulang.
Betul sekali, dan registrasi tahap 2 ini juga sebagai salah satu tahap untuk teman-teman nanti mendapatkan sertifikat dari acara ini. Baik, kembali lagi kita berkumpul di tempat ini dan sebentar lagi kita akan sama-sama mendengarkan pemaparan materi berikutnya oleh Dr. Apoteker Rina Herawati, Sarjana Sains, Magister Sains dari Universitas kita tercinta, Universitas Setiabudi Surakarta.
Baik, tapi sebelum itu Yemima, kita juga akan mengundang moderator kita yang hebat dan tidak kalah dengan moderator yang pertama tadi.
Betul sekali.
Dan berikut akan kami bacakan sekilas perkenalan singkat tentang beliau.
Yap, jadi moderator berikutnya adalah Dr. Apoteker Triwijayanti, Sarjana Farmasi, Master of Public Health, yang saat ini sedang mendapatkan jabatan fungsional sebagai lektor. Beliau lahir di Cirebon dan juga merupakan lulusan dari S1 Universitas Islam Indonesia, Ilmu Farmasi tahun 2007. Berikutnya menempuh pendidikan S2 di Universitas Gajah Mada, Ilmu Kesehatan Masyarakat pada tahun 2009 dan dilanjutkan dengan studi S3 di Institut Teknologi Bandung atau ITB di bidang Ilmu Farmasi tahun 2019.
Oke.
Alright,
now please welcome Dr. Apoteker Triwijayanti.
Tolong aplausnya dong teman-teman.
Oke.
Thank you, MC, for your time. You both look so cute today with your best cap and kebaya, and we feel so traditional. Assalamualaikum warahmatullahi wabarakatuh. Distinguished guests, esteemed colleagues, and participants, it is my great honor to welcome you in the first Setiabudi International Conference, Pharmaceutical and Health here in Surakarta on this amazing day. I am Triwijayanti, your moderator for our last session, the last, of course, but not least. Today, we are privileged to hear from leading experts in our field who will share their insight on significant topic affecting health and pharmaceutical practice. Now, I would like to introduce our keynote speaker for this session, Dr. Rina Herawati. I will read her CV. Dr. Rina Herawati is prominent academic affiliate with the Faculty of Pharmacy at Universitas Setiabudi. She earned her doctorate in pharmacy from the School of Pharmacy ITB and has a Master of Science from the same institution. Her research interests include medical chemistry and biochemoinformatics, focusing on innovative approach to herbal medicine and pharmacotherapy. Dr. Rina Herawati has published extensively in reputable journals and has been involved in groundbreaking research projects aimed at addressing prevalent health issues in Indonesia. There are many publications in Scopus, the newest in silico discovery of potential sodium glucose co-transport 2 inhibitor from Smalanthus sonsifolius via molecular docking and molecular dynamic stimulation approach from Journal of Pharmacy and Pharmacognosy. And of course, a lot of publication in Sinta. And also she has a book chapter and patent. Today, she will present her finding on exploring the anti-arthritis activity of Indonesian herbal medicine. But before we begin, allow me to share pantun. In the morning light we gather here, pharmaceutical minds, let's give a cheers. With herb and science, we'll take a stand to fight arthritis with nature's hand. It makes sound awkward ya, agak krik-krik-krik kalau pantun dibacakan dalam bahasa Inggris. Sehingga saya akan membacakan satu pantun lagi, tolong ya para audiens bisa mengucapkan cakep ya, oke. Naik pesawat lewat bandara, lari-lari sampai berkeringat. Mari kita mulai sajalah acara dengan mengucap bismillah dan tetap semangat. Oke, please join me in welcoming Dr. Rina Herawati to the podium for her presentation. Please give a huge applause. Dr. Rina, the stage is yours.
Thank you, Ibu Dr. Trewi Jayanti, for the nice introduction and the krik-krik pantun. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, all of the participants. 1 p.m. This is a critical hours because our stomach is full and blood perfusion to the brain is minimized. So I hope my presentation will not make you sleepy. Okay. Thank you for the committee for providing me to speak our project result entitled Exploring Anti-Arthritis Activity of Indonesian Herb Medicine. Don't worry because this material is not so heavy, it's so simple. Next. Oh, we can use this. Okay, this is the outline of the presentation. We will start with the introduction about arthritis. The prevalence of arthritis disease, including rheumatoid arthritis, osteoarthritis, gout arthritis, in Indonesia reach among 10% of the population. One of the One of these diseases is rheumatoid arthritis. Rheumatoid arthritis is a common autoimmune disease that is chronic and systemic as well, manifested clinically by polyarthritis associated with a dysfunction of the joint. For you with the age under 30, maybe cannot imagine how is the arthritis. But when, like me, above 50 years, it's very disturbing our daily activity. Rheumatoid arthritis is characterized by synovial inflammation and swelling, autoantibody production, destruction of cartilage and bone, and systemic feature, including cardiovascular, pulmonary, physiological, and skeletal disorder. Let's briefly look at the pathogenesis of rheumatoid arthritis. Pathogenesis of rheumatoid arthritis is quite complex. It involves many factors and many pathways. It starts with the migration of leukocyte from blood vessel to the tissue, and then induce the inflammation in synovial membrane. The next step is auto-activate CD4 T cell. CD4 T cell will activate the macrophage, resulting the production of cytokine pro-inflammatory. Cytokine pro-inflammatory including interleukin 1, interleukin 6, interleukin 17, and tumor necrosis factor alpha. Then this cytokine induce matrix metalloprotein and RANKS ligand production by the fibroblasts. The RANK ligand then activates the osteoclast. We know osteoclast. Osteoclast will deformate the cartilage, and matrix metalloprotein also will destroy the tissues and then resulting the joint destruction. This is what happened in rheumatoid arthritis patient. How about the pharmacotherapy of rheumatoid arthritis? The drug therapy for rheumatoid arthritis was start from analgesic and then non-steroidal anti-inflammatory drug and corticosteroid. But finally, it's reached the revolutionary development of drug. This is the disease-modifying anti-rheumatic drug, or DMARD. DMARDs now are the main therapeutic agents that reduce joint swelling, reduce the pain and systemic inflammation, and of course, reduce the acute phase of markers, so restricting the joint damage. DMARD are classified into two groups. One is synthetic or conventional DMARD. There is methotrexate there. Another synthetic DMARD is sulfasalazine, hydroxychloroquine, and levonomide. In late 1990, the discovery of biological DMARD show the good revolution in therapy of rheumatoid arthritis. Here, the example of biological DMARDs that works as inhibitor of TNF-alpha and inhibitor of interleukin. However, DMARDs and other medication have high cost, it's so expensive, and produce serious adverse effects. Meanwhile, moreover, 30% patients did not show significant improvement when they use these drugs. So, this is a natural product. Indonesia have many medicinal plants that traditionally used for treat rheumatoid arthritis, and natural compound derived from plants such as alkaloid, polyphenol, flavonoid, and terpenoid are great pharmaceutical effect and have the potential to treat inflammatory disease like rheumatoid arthritis. This traditional medicine elicit their anti-inflammatory and immunomodulatory activities by several mechanisms, including antioxidant, radical scavenger, modulation of pro-inflammatory enzyme, and so on. We will focus on 7 Indonesian plants that traditionally used and proved to have anti-inflammatory and anti-arthritis activity. First, we have aloe vera. Aloe vera contain many compounds that show anti-inflammatory and anti-arthritis activity. We also have here ginger or cinnabar officinale. This is clinically proven to have anti-inflammatory and analgesic effect. We also have Pinospora cordifolia or brotowali. Then we have Piper retrofractum or cabai Jawa. Then we have Physalis angulata or ciplukan. We will focus on these 7 medicinal plants. This is the grand design of our project. We conduct anti-arthritis activity in vivo, in vitro, and in silico. In vivo test, we conducted anti-arthritis activity using complete front adjuvant induced arthritic rat, and we measure 2 parameters: ödem volume reduction and the increase of joint space. We conducted in vitro studies, inhibition of TNF-alpha and inhibition of COX-2. We also carried out the in vitro studies. In previous presentasi, Mrs. Nur Ainun have tell you a lot about in silico, and in here I will present our project result of network pharmacology study, molecular docking analysis, then molecular dynamic simulation, pharmacology and toxicology prediction. For in vivo anti-arthritis test, we use Vista rats, which of 180 to 220 gram. We use complete front adjuvant to induce the arthritis. It's injected implantary on the day 1. Then they get booster injection after 7 days. After second injection, the extract were orally administered one daily until 13th day. At the final day, or during this 2 weeks administration, we measure the volume UDIM to obtain the volume reduction of the UDIM. On the final day, the animals were sacrificed and we cut the ankle, the pinhole ankle, and we conducted histology study. This is the resume of mechanism how CFA induce arthritis and Rett model. We know that CFA contain microbacterium. Exposure of this microbacterium will activate, activate migration of leukocyte and so on. Migration of, this is the note, yeah, you can read. The cytokine was released after exposure of the CFA, migration of MPC and dendrit cell, and then proliferation of synovial lining. I'm sorry. TNF alpha, interleukin 1 beta together produce various compound, peptide compound that then promote intracellular addition and migration and so on. With the final effect is hypersensitivity and pain, and the joint swelling. Interleukin 6 also with interleukin 12 and interferon gamma produce differentiation of T cell. Natural killer cell produced by interleukin 12 promote distress in the joint. So we can observe that the animal we induce arthritis get swelling on the joint, and they get quite pain. This is a of COX-2 inhibitory assay. The plant extract first were dissolved in DMSO to obtain various concentration, start from 50, 100, and 200 microgram per milliliter. And then the extract solution was added to immunoassay kit, enzyme immunoassay kit, reagent. There are a buffer solution, then COX-2 enzyme, and the heme. Then the mixture were incubated for 10 minutes at 36.5 degree. After the incubation, we add arachidonic acid. Immediately after adding the arachidonic acid, hydrochloric acid was added to stop the reaction. And then we add stannous chloride to convert PGH2 to PGF2. We use a spectrophotometer to measure the concentration of PGF2 at 418 nanometer, and then we calculate the inhibition percentage of COX-2. This is a scheme I'm sorry. We use THP cells we conducted cell preparation. We use THP cell, and then the extract were added to the THP cell, and we use lipopolysaccharide to stimulate the inflammation in the cell. Then the mixture will incubate overnight at 37 degrees. We centrifuge it, and the TNF alpha quantification was conducted using ELISA. This is the in silico study. Thank you, Mr. Ainun have previously explained about the in silico study. In silico study is, or we can call it computer-aided drug design. We use computer to conduct the calculation. This is an area of computer-aided drug design. We can conduct it virtual screening, compound design, energy of calculation, structure activity relationship, ATME model, and drug target interaction. This is a network pharmacology design, and I see some of the students have conducted network pharmacology design. We collect the natural product or phytoconstituent from the database, from the literature, and so on. And we collect information about target disease, and we conducted the prediction of the target, and this is the quite complex process. And as Mrs. Ainun said before, that we still need validation of result from the in silico study with in silico, in vitro, and in vivo analysis. For molecular docking analysis, we use protein complex we get from protein data bank, and we got some ligands from the database. We conducted preparation of protein and preparation of the ligand. The ligands was converted from 2D to 3D structure. We use ChemDraw and then minimize the energy. Prior docking process, we have to conduct validation to ensure that the docking molecular analysis system is accurate. We use molecular virtual docking, yeah. Then we analyze the rank score or energy binding and the model of interaction ligand and protein. Finally, we conducted a molecular dynamic simulation. Molecular dynamic simulation used to calculate the motion of atom and molecule using Newton's second law of motion. Molecular dynamic simulation is a computational technique that tracks the movement of atom over time. It can be used to study the behavior of biological system, and in this study, it used to measure the stability of the complex ligand and the protein. We also conducted ATME and toxicity prediction, because undesirable pharmacokinetic and toxicology are the major contributor for drug development value. So many web server or software we can use for prediction the pharmacokinetic and toxicity parameter. Here we use AtMatlab 3.0 that based on directed message passing neural network framework. AtMat3 provide a lot of parameters, prediction calculation, 19 physicochemical property, 9 absorption parameter, 14 metabolism parameter, 9 distribution parameter, and so on. Very complex, the parameter they predicted. This is the result of anti-arthritis activity. CFA induced successfully increase the volume of the ankle. We can look at the above picture is a normal joint, and the below picture is arthritis model. All of the plant extract show anti-arthritis activity, but we use two parameter, percent of udem volume reduction and percent of joint space enhancement. We looked at the highest activity on percent of udem volume reduction is shown by Tinospora cordifolia, but the highest activity on joint space enhancement was shown by Physalis angulata. So we use this plan to further study. We also carry out the combination of two plant extract with the 50% and 50% combination. Combination of Physalis angulata and Acalifa indica show the highest activity, and this is in line with the activity of single extract. We can use here that Tinospora cordifolia show a combination with Physalis angulata show highest activity in percentage of udem reduction, but combination of Physalis angulata and Acalifa indica show highest percent of joint space enhancement. This is the histology of hind paw. The left picture is the sample preparation process, and this is the normal group. Normal group show the joint space. Joint space, we measure joint space at 3 points and then average it as joint space distance. This is the arthritis model. We can look that the joint space was very narrow, and the cells and joints area were destroyed. This is the histological result of triamcinolone. Triamcinolone, we use it as a control drug. The distance was wider than the arthritis model. This is the result of group receive Physalis angulata and Acalifa indica. This is the same profile. The narrowing joint space significantly reduced and the cells around the joint space is looks better than at the arthritis model. This is inhibitory activity of COX-2, we can see this is the diclofenac natrium, diclofenac sodium. We use it as a control drug. Diclofenac sodium, diclofenac show IC50 of 38, and this is the IC50 of Physalis angulata extract. The IC50 was 101. Although lower than diclofenac, the activity is quite show enough strong activity as COX-2 inhibitor. This is TNF alpha inhibitory activity. We can see the activity, inhibitory activity 52% obtained on concentration 50 microgram per milliliter. This is a sign of network pharmacology study. The total of 50 compounds from Physalis angulata, and we use HT1 protein target, and there are 16 compounds that correlated with the protein target. This picture is the representation of rheumatoid arthritis protein target. It's obtained from database string. After conducting network pharmacology analysis, this is the simplified profile because we focus on the main protein. The yellow indicate the protein target and the green indicate the phytoconstituent. From a network pharmacology study, we can illustrate the site of action of Physalis angulata. Physalis angulata extract inhibit enzyme COX-2, inhibit PGH2, and so on. Also inhibit pro-inflammatory cytokine like TNF alpha, and inhibit matrix metalloprotein. So the activity of in vivo is relevant with this result of network pharmacology. This is molecular docking analysis result targeting to TNF alpha. Prior to analysis docking, molecular docking analysis, we conducted the validation method and we got RMSD score 1.28 Armstrong, indicate that the method is valid, and the overlay of native ligand from the complex and native ligand from redocking is in similar conformation. This is interaction between native ligand and protein involve methionine 49 and isoleucine 60. This is the docking result of the compound. This is the best three. I'm sorry, I did not show all the compounds, only the best three with the highest rank score. Pisagulin A, farmonolite, and myristin show good rank score compared to the native ligand. And this is the amino acid interacted to the ligand. Native ligand interacted with methionine 149 and isoleucine 50 via steric interaction. Fisagulin A interacted to the protein via this amino acid. Myricetin and farmonolite show hydrogen bond interaction with methionine. This is the same as the native ligand, methionine 149. And the steric interaction also with the isoleucine 70, I'm sorry. This is the model of ligand and protein interaction. This is native ligand and fisagulin A, myricetin, and famonolide. We continue the analysis using molecular dynamic simulation to predict the stability of the ligand and protein complex. We can show that we can analyze that the native ligand, this is native ligand, yeah. Myricetin show a similar model to the native ligand interaction, and its stability until 50 nanosecond. We also conducted ADMET and toxicity profile using ADMET 3.0. This is fisagulin. We can look at some absorption. The green one indicate the parameter is good, the yellow is moderate, and the red is worse. We can look that some parameters are red, but the human intestinal absorption is good, and the permeability of Charcot-2 also good. The distribution almost 50%, health is good. Excretion parameter clearance plasma moderate. This is the metabolism and toxicity. For myricetin show a best interaction to the ligand. Myricetin we use in ADMET profile, myricetin glycoside, myricetin 3-neohexperoxide, because in the extract we found this compound. However, their absorption parameter is not good, but we know that myricetin glycoside will hydrolyze to myricetin in our stomach. This is the conclusion of my presentation. First, all the 7 extract plant, aloe vera, and so on, demonstrate anti-arthritis activity and indicate, as indicated by a reduction of odum volume and the joint space narrowing. Second, the combination of Physalis angulata and Acalypha indica extract show the highest anti-arthritis activity. 3, Physalis angulata extract exhibit COX-2 inhibitory activity with IC50 of 101.16 microgram per milliliter and TNF alpha inhibitory activity. The 3 compound fisagulin A, myricetin, 3-neohexperoxide, and famonolide are predicted the compounds that play an important role in the TNF alpha inhibitory activity. The ADME and toxicity parameter of those 3 compounds varies from moderate to high. This is the end of my presentation. This project was funded by Kemenristek Dikti, and thank you for all the teams that support this project. The time for the beautiful moderator. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Dr. Rina Herawati, for the presentation. Please take a seat. We now open the time for discussion. I encourage all participants to engage actively by raising your question and sharing your insight. Please state your name and affiliation before posing your question. And the same like the first, yeah. Please feel free to raise your question. Is there?
Thanks for the time given to me. My name is Indriana Wulandari and I am a student at Setiabudi University. What I would like to ask is, I have a family member who suffers from rheumatoid arthritis and regularly drink boiled salam leaves. My questions, are there any side effects associated with consuming salam leaf infusion? And are there any physical exercise that can help alleviate the discomfort? Thank you.
Thank you for the question, Mbak Indriana. Now I would like to invite our speaker, Dr. Rina Herawati, to address these inquiries. Dr. Rina, please share your insight.
Okay, thank you Mbak Indriana for your question. Salam leaf was reported have side effect hypotension. So for the chronic treatment and for the patient with low blood pressure, it's quite need warning. But if your family have no problem with blood pressure, I think it's no problem. Yeah. But salam leaf, the side effect of salam leaf reported was hypotension. For the reduction the pain on the joint, you can use compress, yes, compress with warm water shock maybe to reduce the inconvenience or the pain usually in the morning and the evening, or use topical agent from our project. Always administration significantly reduce the inflammation and the pain. The ginger, jahe, also significantly reduce the inflammation. But the result I'm not reported the result of using the topical administration of aloe vera and ginger, but it significantly reduced the pain. Is answer your question, Mbak Indri?
Thank you, Bu Rina, Mbak Indri ya. Jadi nanti bisa dikompres pakai air hangat. Okay, the second one. The second question, yes? You?
Yes. Good afternoon. Let me introduce myself. My name is Tasya Yunarti Wardani, and I'm student University Setiabudi. And my question is, given the promised result of pengangulata extract in Burina study, what are the next steps you recommend for translating this findings into clinical application for managing rheumatoid arthritis? Thank you.
Okay, thank you Mbak Tasya. Actually, we have finished our project and we have get the composition of 3 plants administratively and one plants topically. But it's from the in vivo study and at Redmond. We have not been conducted the assay on human because it is quite difficult in Indonesia to in clinical assay, but our formulation content of 3 plants and 1 topical we use aloe vera for the administration. Actually, we will continue this project because we got topical administration, and for the deep joint, it's need a delivery system to make the beta constituent reach the site of action. So we plan to collaborate the colleague from technology to design nanoparticle formulation to enhance the penetration of the substance reach the site of action.
Thank you. We still have 10 minutes left. Maybe 1 or 2 questions. Yes.
Good afternoon. Thank you for the opportunity. My name is Alexandra Jenia Sinta. As I am student at Universitas Setiabudi. Specifically, I am a thesis advice of Ibu Triwijayanti. What I would like to ask is, what challenge do researchers face when integrating traditional herbal medicine into modern pharmacotherapy for arthritis, particularly regarding standardization and dosage? Thank you.
Okay. Thank you, Mbak Alexandra. You're right that this is still the challenge for rheumatoid arthritis therapy with natural product because we have biological agent demand that the efficacy is superior compared to traditional product. So the traditional medicine, traditional product is not the main therapy. It's only a adjuvant or add-on therapy for rheumatoid arthritis because the progress of the disease is very fast. So from our research, it's difficult to get the same activity with the DMAT or biological DMAT. So yeah, adjuvant therapy, not the main therapy.
Okay, maybe the last one, the last chance. Yes, it will be.
Yeah.
Assalamualaikum warahmatullahi wabarakatuh. My name is Rizky Kamila Permata. I am from Universitas Setiabudi. My question, what factor cause approximately 13% of RA patients to show no significant improvement with existing pharmacological treatments? Terima kasih. Thank you.
Thank you, Mbak Rizkita, for the question. The failure of treatment therapy with DMAT, one is because chemical DMATs or conventional DMAT need long time to post the activity. Onset of actionnya, the onset of action about 1 month. So some patient feel that the drug is not used for treat the rheumatoid arthritis is the first. Or because this drug have to consume every day, so the compliance of the patient is important role in the successful of the treatment. about biological DMARD, the side effect is infection. So if the patient use biological DMARD, the side effect infection could happen, and automatically we must stop the therapy. That's meanwhile the progress of immune dysregulation is still is still in immune dysregulation. It's happen in our body. So the therapy for rheumatoid arthritis is up and down for the patient.
Okay, thank you. I know you have a lot of questions, but the time is up. So as we conclude this session, I would like to summarize the key point discussed today. Dr. Rina Herawati highlighted the prevalence of arthritis in Indonesia and emphasized the potential of natural compound derived from Indonesian herbal medicine as effective treatment for arthritis, particularly through their anti-inflammatory properties. The findings presented today underscore the importance of further research into herbal remedy as viable alternative or adjuvant to conventional treatment for arthritis. In closing, I would like to express my sincerely gratitude to Dr. Rina Herawati for her valuable contribution and insight today. Let us give her a round of applause for her exceptional work. Before I return the program to the Master of Ceremony, I still have one pantun and I will deliver in Indonesia. So you know what you have to do? Cakep, okay. Buah nanas, buah koweni. Dimakan bersama ibu terkasih. Cukup sekian sesi hari ini, saya ucapkan banyak terima kasih. Have a wonderful day.
Ladies and gentlemen, the great material by our last speaker has ended. And now it is the certificate submission by Apoteker Dian Marlina, Sarjana Farmasi, Magister Farmasi, Master of Science, Doctor of Philosophy, as Chairman of the Committee.
Hadirin sekalian, tiba saatnya adalah pembagian sertifikat, penyerahan sertifikat oleh Apoteker Dian Marlina, Sarjana Farmasi, Magister Sains, Master of Science, Doctor of Philosophy selaku Ketua Panitia. Kami persilahkan.
And to our speaker, we also welcome to you stand up and receive the certificate.
Berikutnya adalah penyerahan sertifikat kepada moderator keren kita, Ibu Dr. Apoteker Triwijayanti. Terima kasih Ibu sudah memandu acara kita hari ini dengan sangat baik. Wow.
Wow.
Banyak sekali fansnya ya.
Iya.
Waduh, semoga ketularan ya seperti beliau. Baik, inilah sesi foto bersama antara moderator, pemateri kita dan juga ketua pelaksana. Nah, setelah ini,
ya, dengar-dengar tadi ada yang membalas pantunnya Bu
iya,
Triwijayanti.
sebentar lagi kita akan mendengarkan balasan pantun dari Bapak Dr. Apoteker Iswandi, Magister Farmasi. Nah, mungkin langsung saja Bapak Iswandi, kami persilahkan untuk membalas pantun dari Bu Yanti nih. Monggo Pak. Ya, so Dr. Apoteker Iswandi will play Dr. Apoteker Triwijayanti pantuan.
Ya.
Oh masih
Iya, kayaknya ini pantunnya sangat bagus sekali.
Jangan lupa cakepnya ya teman-teman.
Ya, cakepnya yang keras ya teman-teman.
Aduh, Pak Dekan udah mau pantun ini. Request by apoteker Dian Marlina ya Pak, bukan dari kami ya Pak. Iya.
Cakep ya? Oke.
Cakep.
Bismillah. Saya balas pantunnya. I want to repeat the pantun from the. Oke. Sebentar lagi inspirasi dulu. Anak gadis pergi ke KUA. Tidak lupa membeli terasi. Usai sudah acara kedua, terima kasih semua partisipasi.
Luar biasa sekali pantun dari Dekan Universitas.
I think I don't come to speak pantun or the Dr. City. I can speak pantun. Oh no. Oke, monggo kembali lagi.
Ya, sangat seru sekali ya balasan pantun hari ini. Anak gadis pergi ke KUA.
Ya, cakep.
Tadi lupa.
Oh,
Maaf ya
tak
Pak, saya lupa
mau
ya Pak.
balas pantunnya Pak Iswanti tadi.
Dan mungkin sekali lagi kami ucapkan terima kasih banyak atas materi yang telah diberikan oleh Dr. Apoteker Rina Herawati dan juga yang telah memandu acara kita hari ini, Ibu Dr. Apoteker
Triwijayanti. Berakhir sudah rangkaian kegiatan utama kita dan sesaat lagi kita akan sama-sama masuk dalam sesi parallel session oral presentation.
Yes, our main part of activities has ended and in the moment we will enter the parallel session oral presentation.
Nah, tapi sebelum memasuki acara parallel session oral presentation nih, aku mau ada info sedikit untuk para peserta seminar.
Apa nih?
Nah, tadi masih kenyang?
Udah lapar.
Sudah lapar. Nah, pas sekali karena ada sesi coffee break.
Jadi untuk coffee break sendiri nanti akan berlangsung selama
Ya, dimulai dari sekarang sampai 14.15. Jadi untuk para peserta yang ingin mengambil coffee break akan dibuka jam sekarang sampai nanti. Dan kalau mau mengambil, saya himbau untuk jam 14.15 untuk segera kembali lagi ke sini.
Iya, boleh dimakan di
Ya.
sini boleh, seperti tadi pagi.
Ya, langsung dibawa ke sini ya, teman-teman.
Kalau yang ingin masih kenyang lah, nanti bisa diambil nanti.
Ya, coffee break ada di depan gedung ballroom ini ya, pas ya kayak ada warung itu, nah itu coffee breaknya.
Dan jangan pergi dahulu ya teman-teman, karena nanti setelah ini akan ada presentasi
Session oral
Iya.
tambahan dari pembicara kita yang tidak kalah hebatnya.
Ada dari Dr. Siti Nurfadzilah binti Musai
Ya.
from University Teknologi Mara yang sebentar lagi akan memberikan kita sedikit materi
Betul sekali.
dan nanti akan disampaikan oleh moderator dalam ballroom ini oleh seksi ilmiah. Ya mungkin untuk teman-teman sekalian yang ada di rumah ini bisa nih juga istirahat dulu.
Ada informasi juga yang tidak kalah pentingnya untuk peserta parallel session oral presentation akan dibagi menjadi 4 ruangan. Di ruangan di lantai 2 ada Meru, Pamiluto, dan Kawung. Bagi peserta yang menempati ruangan Meru, Pamiluto, dan Kawung ada di lantai 2 di depan resto, dan di Sidomukti ada di lantai 1 depan resto. Bagi peserta para session oral presentation bisa segera untuk menempati ruangannya masing-masing.
Iya, ada 5 ruangan dan salah satunya ada di tempat ini.